Warm welcome to all the attendees and audience. It's heartening to withstand the energy and enthusiasm pervading the hall. Let us begin the event with an auspicious note of mellifluous fluid recital invoking Vignaharta Vinayaka Mahabganapatim Bhaje by our in-house flutist Bhuma Mahendra. Welcome Bhuma Mahendra. Thank you, Mr. Mahendra. That was mesmerizing and melodious performance. Now we'll move on to the today's next program. We have Sangeet Nritya Sangamam, an ancient classical dance symbolic to Telangana's culture and heritage by Kumari Mahanya and Kumari Varunya, Penini Natyam.
మంజుల శింజిని చతుల లాస్య వినోదవ శంకర
Pali
Thank you, Mahanya and Varunya, for such a rhythmic and graceful performance. Next, we'll now begin the proceedings. We respectfully invite our principal, Dr. Ajay Kumar Garu, to grace the dais. I request Uma Maheshwari to present the bouquet to Principal Sir. It is an honor to invite Dr. Sumanlatha Ma'am, BOS Chairperson of Genetics, Osmania University, onto the dais. I request Shivani to present the bouquet to Madam. Request Dr. Anuradha Garu, CEO and founder of GeneTech Laboratories, Hyderabad, to grace the dais. I request Diti to present the bouquet to Madam. It is our privilege to welcome Padma Shri Awardee and CSIR-Shanti Swarup Bhatnagar Fellow, CCMB Hyderabad, Dr. Kumaraswamy Thangaraj Garu, to adore thee and enrich the dais. And I request all for a round of applause. I request Kaveri to present the bouquet to Sir. Sciences and continues to the dynamic and rapidly advancing field that impacts everyday aspects of life, from the intricacies of the inheritance and the molecular dance of gene expression to far-reaching the implications of genetics in medicine, forensics, agriculture, and evolution. Today's talk focuses on key current and emerging trends in the genetics. Now moving on to Now I request all our esteemed guests to illuminate today's proceedings by lighting the holy lamp.
कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुध्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्परब्रह्म दीपज्योतिर्जनार्दनः दीपज्योतिर्परब्रह्म दीपज्योतिर्जनार्दनः दीपो हरतु मे पापम् सन्ध्यादीपनमोऽस्तु ते ओ दीपब्रह्मरूपस्त्वं ज्योतिषां प्रभुरव्ययः ओ दीपब्रह्मरूपस्त्वं ज्योतिषाम् आरोग्यं देहि यावत् सूर्योदयो भवेत् रवेरस्तं समारभ्य यावत् सूर्योदयो भवेत् यस्य तिष्ठेत् गृहे
Thank you so much, madam. Now I shall hand over to Isra Mohammed to introduce our eminent speaker, Dr. G. Sumanlatha Madam, BOS Chairperson. Over to you, Isra.
Sumanlatha Garu has obtained her PhD in immunogenetics from the Osmania University in the year 2002. With over 20 years of research experience, she has presented her work at several national and international conferences and has published in reputed national and international journals. She was selected for the scheme of Young Scientist and for the scheme of Young Investigator by the Government of India. She has served as the principal investigator and co-investigator in multiple projects funded by DBT, ICMR, DHR, and DST. Her research specialization includes immunogenetics of asthma, tuberculosis with diabetes, and diabetic nephropathy. Dr. Sumanlatha has presented her work at various international forums, including Giza and Siva Oasis in Egypt, the Freie University Berlin of Germany, and she is also a member of Indian Immunology Society and Indian Society of Human Genetics. She was a member of Indo-German International Research Training Group, IRTG GRK 1673, on functional molecular infection epidemiology from 2009 to 2015. Dr. Sumanlatha currently serves as a co-editor of Frontiers in Immunology. We are pleased to invite Dr. Sumanlatha Garu to deliver her address.
Faculty and the students, good morning to all of you. First of all, I would like to thank the convener of this program, Ms. Padma, Head Department of the Genetics, and also the principal of this college, Aurora Degree and PG College, for giving me an opportunity to be part of this program. All know that tuberculosis, which is one of the infectious disease, which has been distributed all over the world. And this is one of the leading causes of the death from this bacterial infection. Earlier in the 17th and 18th century, this TB was known as a white plague, and it has been there in the Europe. Like 100% of the people were infected with this, and 25% of them were died due to the disease. But later in 1882, Robert Koch, a German microbiologist, who identified Mycobacterium tuberculosis as one of the etiological factor which is responsible for TB. Next slide. Yeah. So in 1990, it was going on. So in 1990s, the global TB epidemic has worsened due to the factors like using of the emergence of the drug resistance TB, use of the immunosuppressive agents, drug addiction, poverty, and the population movement. So these are the factors which are trying to hinder the control of the TB. So this has again become a re-emerging infectious disease where it has become a serious global health problem, especially in the developing countries. So this pulmonary, this TB. This is of two types. Like this has been, if it affects in the lungs, it is called as a pulmonary TB. If it affects other parts of the organs of the body, then it is called as a extrapulmonary TB. So if you look into the global estimates of the TB, it has been reported that 10.7 million people fell ill with TB and 1.23 million died from the disease. But if you look into the incidence rates of this TB, it is 131 for the 100,000 population, and the case fatality was 11.5%. So most of the people who developed TB were in the 30 high burden countries. Among them, if you look into it, so the top 8 countries which has been reported by WHO in 2020-25, it is the topmost is the India, which is with 25%, followed by Indonesia, Philippines, China, Pakistan, Nigeria, the Democratic Republic of the Congo, Bangladesh. So if you look into the gender-wise also, male, female, the males were more compared to that of the females. That is the 54% of the people were developed TB in males, 54% of them are males, and 35% were females, and 11% were children. So this has become an emergence, or it is required to end the global TB epidemic by 2030. So this has been, not only it has been adopted by WHO, it has also been adopted by all members of the United Nations, to which they all want to be see that it should be controlled by 2030. So this is again a latest report where they have compared between the 2015 and 2025 the estimates. So the TB incident rate has been reduced 50% reduction by 2025 compared to that of the 2015, and the death rates also there is a 75% reduction, and even the contacts where there is no cost effectiveness is there for them. So finally, it has been decided by WHO and the United members they have committed to end the global TB epidemic through their adoption of the End TB strategy and the SDG goals, where they have decided, they are expecting that by 2030, the targets of the End TB strategy should be a 90% reduction in the number of deaths caused by the TB and 80% reduction in the TB incidence. So if you look into the natural history of TB, here you can see that, so whenever a person gets exposed to the TB, within 4 to 6 weeks, there is a possibility of the elimination of the bacteria. If the bacteria has not been eliminated, then it may lead into the active TB. And if the initial immune system are trying to control the bacteria, they may go into the latent TB form. And this latent TB form, where in the lungs the granuloma formation will be there, where the TB bacilli will be in the dormant stage. So this latent TB will be there forever throughout the life, or it may get eliminated, the bacteria may get eliminated, or the person may develop a reactivation where it may turn into the active TB. So if you look into the symptoms of the TB, they'll have a persistent cough for lasting 3 weeks or longer. Potentially the cough will be with blood or the mucus, chest pain during the breathing or the coughing, and the systematic symptoms like high fever, night sweats, chills, and they'll have a feeling, generally they'll be unwell, and the loss of appetite and the weight loss will be there, and they'll have a fatigue and the weakness. And if you look for the symptoms of the extrapulmonary TB, they'll have a lymph nodes swollen and firm, they'll be painful and glands in the neck and the armpit, and the meninges, that is a TB meningitis, where they'll have a headache or the confusion or the stiffness in the neck. Spine, if they have infected with TB to the spine, they may have a back pain or a stiffness, and if it is affecting the kidneys, they'll pass the blood in the urine. So if you look into the treatment, so there are different forms of the treatments, like DOT strategies are also there. So here you can see the active pulmonary TB treatment can be done in 6 months in 2 phases. one is the intensive phase, the other one is a continuous phase. So the intensive phase is it will be there for first 2 months of the treatment with a combination of isoniazid, rifampicin, ethambutol and pyrazinamide, and the continuous phase will be there for 4 to 7 months with isoniazid and the rifampicin. So these are the drugs, how they will be affecting, and these are the first line anti-TB medications. And apart from the patients, the contacts are also like who are being getting exposed to the patient, they are also given a preventive therapy with isoniazid for 6 to 9 months or rifampicin for 4 months to prevent the development of active TB in the household contacts. So not only the exposure, there are risk factors also which are trying to play a role here. So the risk factors here, if you see, both the host genetic factors and also the socio-economic factors are also trying to play a role in the susceptibility and resistance towards the disease development. So the host genetic factors are, like we can see, the age, gender, immune status, malnutrition and the diabetes, and the environmental and the social factors, socio-economic factors, if you see, overcrowding, poor ventilation, alcohol, smoking, and the occupation is one of the risk factors. So these are the risk factors which are there for developing a resistance or susceptibility towards the disease. Not only the risk factors, even the comorbid conditions are trying to hinder the outcome of the disease, like if the person is suffering from HIV along with the TB and also the diabetes, and other conditions like the chronic lung diseases, cancer, kidney diseases are the mental health disorders. Okay, these are trying to hinder the outcome of the TB. So now these are the symptoms, treatment, and how they are, what are the different risk factors. Now we are saying not only the TB patients, even the household contacts are going to develop a disease. So among the global population, if you see, so 30% are exposed to MTB and they are infected, and which is leading to 5 to 10% active TB. So each active TB individual is trying to infect 10 to 15 individuals annually. So here we are going to concentrate more on the, in my study, more on the household contacts. Household contacts are at a higher risk of developing a TB than the general population because of the due to the predisposition towards the genetic factors or the environmental factors or the transmission of the infection. So we are saying that genetic susceptibility, yeah, genetics is trying to play a role, not only the genetics and even the ethnicity is also trying to play a role here. So the occurrence and susceptibility to TB vary significantly across, we know that different ethnic groups, regions and the population, and these disparities are largely attributed towards the host genetic factors. So especially it could be a variation in the gene specific frequencies or even the environmental variables like the penetrance and the selection criteria, where all they are trying to influence these genetic traits which manifest in the different population. So here we are talking about the genetics. So I am more interested in the cytokine gene polymorphisms. So these cytokine gene polymorphisms are, they are the single nucleotide polymorphisms in the cytokines. They are the primary determinants of the individual's immune responses towards the TB. So here, if you see these polymorphisms, they're not only initially susceptible, but the clinical severity and the long-term outcomes also can be known by the genotypes. Okay, so these cytokines, they will try to tell us whether a host mounts, whether it is of Th1 or a Th2 type of the immune responses upon exposure to the TB bacilli. Okay. So now this is the pathogenesis, how here I am just going to tell you about how the innate and adaptive immunity is trying to activate when a person gets exposed to the MTB bacilli. So whenever a person gets exposed to the, like when the MTB gets exposed, we get exposed, first they will move towards the pulmonary alveoli through the respiratory tract. So initially this bacilli will be recognized by the resident cells of the innate immune responses like the macrophages and the natural killer cells and the dendritic cells. So here the dendritic cells will, the immature dendritic cells will recognize the bacilli at the infection site, and then they move towards the lymph nodes where the immature dendritic cells gets differentiated into mature dendritic cells. So the mature dendritic cells will try to increase the activation of the antigen presenting cells, and also they try to enhance the expression of the class 1 and class 2 MHC molecules. So these class 2 MHC molecules, they try to activate in the presence of CD40 and 40 ligand the CD4 T cells. These CD4 T cells, they further get differentiated into different subsets like Th1, Th2 and the Th2, Th17. So based on the environmental cytokines, they try to contribute towards the control of infection. Here, both the Th1 and Th2, they try to maintain homeostasis of the immune responses. And class 1 MHC, they try to in the presence of CD28 and CD80, they activate the CD8 T cells where they try to differentiate the cytotoxic T lymphocytes to release the granzyme perforin fast ligand or gamma TNF alpha, where they try to activate the macrophages to eliminate the bacteria. So depending on the interplay of the innate, adaptive and the virulence of the MTB, the infection can be either it can be recovered, or it may go into the latent TB form, or they may develop into the active TB. So these are the immune response markers which I have studied, we study in our lab, and these are the MTB ligands, antigens, mycobacterial antigens, recombinant cytokines, cell surface markers, apoptotic markers, and different pro, anti and the regulatory inflammatory cytokines which we study in our lab. Okay, so today I am going to concentrate mostly on the pulmonary TB patients, relapse patients and the household contacts. So the pulmonary TB patients were defined as they are sputum smear positive, and sputum will be positive for the acid fast bacilli. And the relapse cases are the patients who have already completed their treatment, but they have developed the disease. And the household contacts, the definition of the household contacts is an individual who has resided in the household for at least 7 consecutive days during the 3 months prior to the diagnosis of the tuberculosis patient. Mostly we see in the household context, mostly they will be the siblings or the spouses. So now I was telling you the diagnosis. The diagnosis is based on the sputum smear examination for the acid fast bacilli, sputum culture and the chest X-ray as per the RNTSP guidelines. Okay, so for these patients and the contacts, we try to do the tuberculin skin test positivity to check them, and also we'll calculate their body mass index, and all the studies which we are carrying out, they'll be approved by the institutional ethics committee, and also the patient's willingness, whether if they are willing to give a consent only, we'll be including in the study. And the exclusion criteria is the people who are having a comorbid conditions like HIV disease or a diabetes, hypertension, renal transplant, or cancers and the cardiac patients, they are all were excluded. But here in each study, the number of subjects in my study are trying to vary. So whatever the methodologies which we carry out in our study, in our lab, I'm going to study mostly on the TB patients, household contacts and the healthy controls. So these are the different techniques which we carry out. That is a lymphoproliferative assay, cell culturing, ELISA, flow cytometry, and the molecular work like PCR and the RT-PCR. So today I am going to talk on these aspects: the innate immunity, the adaptive immunity, the recombinant cytokines, and the apoptotic markers. So these are the contents which I am going to touch today. So first is I am going to tell about the importance of the MTB antigens. So the MTB antigen, that is the antigen 85A, which is a structure, it supports the structural integrity and the unique composition of the cell wall, where it is very important for the MTB ability to evade the host immune responses. Okay? So upon infection, what happens? The immune cells present the antigen 85A2 to the T cells, where they start initiating the immune responses. And if you talk about the ESAT6, this is an early secretory antigenic target 6 kilodalton protein, antigenic protein, which is secreted by the bacterium, and it is one of the part of the ESX secretion system, which is essential for the virulence of the MTB. And its primary function is to disrupt the host immune responses by lysing the host cell membrane and facilitating the MTB to escape from the phagosome into the cytosol of the macrophages. And I am going to talk about the ligand. Ligand, this is one of the ligand, which is a PAM. The PAM, which is a synthetic tripalmitoylated lipopeptide, which acts as a potent toll-like receptor agonist, where it is very important for the initial immune responses to activate the macrophages and promoting the cell proliferation. So it is one of the which is used in the research to study whether the NFK beta pathway has been activated or not, to see its vaccine efficacy and also to see against the protection against the bacterial and the viral infections. So now I am talking about the host pathogen interactions. To activate the host defense mechanism, here it involves both the innate and adaptive immune system. So the innate immune system, it depends mostly on the recognition of the several pattern recognition receptors which are present on the phagocytic cells, in which the toll-like receptors are the ones which are trying to play a major role in the innate immunity. So if you see the mechanism of the toll-like receptor signaling pathway, so whenever a PAM or a ESAT6, when they bind to the toll-like receptor, this is a ligand, this is an antigen. When they bind to the toll-like receptor, this TLR2 forms a heterodimer either with a TLR1 or a TLR6, and this is an MYD dependent pathway where they try to recruit the adaptor molecules for the phosphorylation of the different kinases, and ultimately they try to activate the MAP kinase and the NFK beta pathway. So here in once the activation of the NFK beta pathway occurs, there is a transcription of the genes takes place where they try to produce the pro-inflammatory cytokines. So I have studied the PBMCs of the patients, contacts and healthy controls, where we have stimulated those PBMCs with the ESAT6 antigen, and we have seen and we have followed the patients from 0 month to 24 months. How their immune responses are there, not only the patients, even the household contacts also we have followed to check their immune status. So here we have seen that, and here in the contacts are being divided into, we have tested the contacts with the gamma interferon. Gamma interferon is a test where it will tell you whether the TB infection is there or not. So we have divided the contacts into two groups, positive to the quantiferon and negative to the quantiferon, and we have divided. So here initially the low pulmonary TB responses were there, and they have been increased as the treatment has been started, and there are similar results we have observed in the contacts where there is no difference whether they are quantiferon positive or negative. And again we have stimulated with the PAM, that is a ligand. Here also we have seen that the people with quantiferon positive, they have a low immune responses, but they have enhanced with the treatment, but there are dissimilar results with the contacts, though they are significant, but if you see how they are increased, there is a dissimilarity in the household contacts. And even the quantiferon negative, here we have seen there are low in the PTB patients, and then they have decreased with the immunity, and they have increased their immune responses during the follow-up. And as I was telling that the pro-inflammatory cytokines will be released when there is a binding of the TLR2 or a PAM or a antigen with the TLR2, there will be a release of the pro-inflammatory cytokines. We have checked the pro-inflammatory cytokines of IL-1 beta, TNF alpha and the IL-12. So the IL-1 beta is the one which promotes the antimicrobial immunity in macrophages, and it is one of the key mediator of the inflammation, where it plays an important role in the host resistance to Mycobacterium tuberculosis infection. And TNF alpha, it also stimulates to migrate the immune cells to the infection site, contributing to the granuloma formation, where they are capable of controlling the disease progression. So usually we see the high levels of TNF alpha which are being detected in the culture supernatant of the patients who are being treated with, who are being stimulated with the mycobacterial antigens. Okay? And IL-12, which is also very important for the innate immune responses, resistance to MTB infection, and this IL-12 is essential to generate a protective immune response towards the MTB, which has its main function being the induction of the expression of the IFN gamma and also the activation of the antigen specific lymphocytes, which are capable of creating a protective granulomas. Okay. So again we have estimated these levels also in the culture supernatants. The cells which are being stimulated with the MTB antigen, in their culture supernatants we have estimated the release of pro-inflammatory cytokines. So we have seen that in the quantiferon positive, the high levels of IL-1 beta were there initially and then they have increased in patients, but there is no much difference in the during the follow-up in the patient, in the household contacts. In the quantiferon negative also we found that high levels of IL-1 in both the patients and household contacts, but they have decreased during their follow-up. So what could be the reason? Exactly we do not know. This is how we got it. But whereas the TNF alpha, both in the quantiferon positive and negative household contacts, it is very low and slowly it has been increased during the follow-up. And IL-12 also we see that they have increased initially and then decreased at 6 months and then again they have increased at the 24 months. And in the quantiferon positive household contacts, if you see in this group, they are low in the initially both in the patients and the household contacts, but they have increased during the follow-up of the 24 months. And we also estimated the these cytokines stimulated with the PAM antigen or PAM ligand. Here also we can see that there is a high pulmonary TB, they are high in the pulmonary TB and decreased with the treatment. So there is no difference both in the quantiferon and positive and negative. The household contacts we have shown gradually decrease in their levels during the follow-up. And TNF alpha also, they are in this, the TNF alpha in both the patients and contacts during the follow-up, it has been increased gradually from 0 month to 24 months. And IL-1, IL-12 in the culture supernatants, if you see, the quantiferon positive, low in PTB and high in SSC, first they have increased and then they have decreased in both the patients and contacts. And quantiferon negative, if you see, they are initially low in PTB, but there are similar in household contacts during the follow-up. So even not only the ELISA levels we have estimated, even we have carried out the mRNA expression also. Okay, so we have seen for the cells which are being stimulated with the ESAT6 antigen and also with the ligand PAM. So here we have estimated the TLR2 expression. Here, so we have stimulated, this is a PAM stimulated cells. The stimulated cells with PAM were significantly low in the patients when compared with the healthy controls by 2-fold, while the household contacts did not show significant difference in the mRNA expression during the follow-up with the PAM. But whereas the mRNA expressions, if you see with the ESAT6 antigen, here they are significantly low in PTB and household contacts by 8-fold when compared to HC. But during the follow-up, the expression is increased in pulmonary TB, while it is decreased in the household contacts at 24 months when compared to that of the 0 month. So finally, if you conclude, if you see all this data, the conclusion of this is in vitro stimulations which we have done with the TLR2 and the PAM ligand and ligand of the PAM, TLR2 and also ESAT6, they have shown a decreased TNF alpha, IL-12 and increased IL-1 beta, where it is increasing, indicating that a high inflammatory reaction and protection against the TB in the household contacts. And the results, if you see on the whole, it has been concluded that the pulmonary TB patients, they are experiencing a broad multi-level recovery of their immune system, like the cellular, cytokine and the genetics, which are trying to play a role during the following of the treatment. And the household contacts, they are exhibiting a more complex and sometimes they are worsening the immune profile despite not having the active disease. Though they have an infection, they are not developing the disease. They are in between the patients and the controls. And also we have carried out the SNP analysis for the TLR2 at two positions like TLR2 2029 C2T and TLR2 2258 G2A polymorphism. These are the gel pictures of the two positions. And if you look into the genotypes of the TLR2 2029 C2T gene polymorphisms in patients, contacts and healthy controls, we have observed that C2T, a codominant, was susceptible in the TB patients compared to healthy controls, and TT, the recessive in patients and the household contacts, and the CT of the dominant and the codominant model have shown the protective in the household contacts. And other SNP which we have looked into it, other position of the TLR2, that is 2258 G2A, here only the over dominant was found to be susceptible in the patients when compared to that of the TB healthy controls. So the conclusion of these SNP analysis is the TLR2 2029 C2T variant might be a robust biomarker for tuberculosis outcomes compared to that of the 2258 G2A variant. And here if you see the allele wise, the T allele in the 2029 position serves as a critical protective factor that may prevent the transmission from exposure to the active disease, whereas the 2258 G2A variant, it tries to increase the vulnerability towards the disease. So this has been published in the paper Cytokine, all this data. And now we will be switching on to the adaptive immunity. Adaptive immunity in the host pathogen interaction, I am going to talk only about the IL-4 and the TGF-beta. So IL-4, which we all know that it regulates differentiation of antigen activated by the naive T cells, where it tries to exhibit the Th1 cytokine suppression. And upon when the person gets exposed to the MTB, where there is a possibility of there is an increase in the IL-4 responses, and that tries to down-regulate the Th1 levels, which results into the failure of the protective immune responses with the high IL-4 levels. So studies have suggested that effective vaccine development is for blocking the IL-4 responses is required than inducing the Th1 responses. Okay? And this cytokine is present on the long arm of the chromosome 5 at 31.1 position. And the functions here we see that the functions of IL-4 is mixed, like they have a mixed Th1 and the IL-4 responses, where it is leading into the failure of the Th1 cell mediated immune responses towards the TB. Okay? And another cytokine which I am going to talk is the TGF-beta. TGF-beta, which controls the immune system and demonstrates diverse activities on different cell types and cells at different developmental stages. So this TGF-beta, which is present on the long arm of the chromosome 19 at 13.1 position, and if you look into their towards the immune responses, this TGF-beta acts as a critical brake on the immune responses, helping to maintain the self-tolerance and also prevent the excessive inflammation. So we have looked for the lymphoproliferative responses against with the MTB antigen, and here we have seen that low mean proliferative responses found in, we have seen the adaptive immunity not only in the active pulmonary TB patients and also in the relapsed patients and the household contacts of both the categories and also the healthy controls. Okay, so the low mean proliferative responses were found in the APTB patients, relapsed patients and their household contacts when compared with the healthy controls. And even this is another group where TB patients and their household contacts, if you see during the follow-up from 0 month to 24 months, the mean proliferative responses increased during the follow-up both in the patients and the contacts, and this is a group of the relapse and their household contacts. Here also we see that there is an increase during the follow-up. So there is in both the groups, that is APTB and the relapse cases and their household contacts, the similar observations were observed. Okay, and we have estimated in the stimulated cells in the culture supernatants the IL-4 levels. The mean IL-4 levels, if you see, they are found to be in APTB, relapse and the healthy controls were compared with the healthy controls, and here we see that the mean stimulated levels decrease during the follow-up from 0 to 24 months, and in the relapse also there is a decrease. Both the stimulation index and the levels were trying to decrease during the follow-up in both the categories. And TGF-beta levels, Here we have seen that the mean stimulated TGF levels decreased also. Here also TGF beta levels were decreased with the MTB antigen 85A. Okay, so the conclusion of this adaptive immune responses is the baseline elevation was there both in the TB patients, relapse and their household contacts, where they are showing significantly higher mean levels of IL-4 compared to that of the healthy controls. And during the follow-up, we see there is a decrease in the IL-4 levels, both the categories, both the groups, that is APTB and the relapse cases, and also in their healthy controls. So TGF beta levels also were significantly, initially they are significantly higher in APTB and the relapse patients in the healthy controls. And during the follow-up, also these cytokines level also has been decreased both in the relapse cases as well as in the active pulmonary patients. So on the whole, what we try to conclude that the host genetic variations and specific cytokine profiles are the major determinants for the TB susceptibility and also for the severity. So if you monitor the decline of these suppressive cytokines, will help us out, will try to help us for tracking the clinical outcomes and also developing a novel prevention strategies. So same thing in the IL-4 and the TGF beta, we have carried out even the SNP analysis. So at two positions for IL-4 at my 589 position C2T and the IL-4 C2T minus 33 position, we have done the SNP analysis in the patient's contacts and the healthy controls. Here we have seen that only the CT genotype is protective in the APTB patients, and also based on the AICT, it has been reported that CT is protective. CT genotype is protective in them, and here there is no significance for this position. And in this also IL-4 minus 33 C2T position in the APTB, it has not been significant, and in the relapse also it is not shown significance. And the TGF beta also we have carried out two positions, the SNP analysis. One is T2C plus 869 position, another one is a C2T minus 509 position, and we have seen that the APTB and the APTB household contacts versus healthy controls, where the dominant model TC was found to be protective, and the similar results and dissimilar results were reported by the different population here. And the TGF beta, another in the relapse cases, also here also we are able to see the dominant CT genotype was found to be protective, and TGF beta another position minus 509, where we are able to see that TT genotype was found to be protective in the APTB patients, and in the relapse also the same genotype was found to be observed. And in conclusion, in summary, if you see the adaptive immune system, the IL-4 minus 589 C2T variant, it primarily influences the initial defense against the active infection, while the IL-4 minus 33 C2T variant serves as a critical predictor for the disease recurrence. Okay. So finally, what we conclude from this SNP analysis is there is a specific variation in the TGF beta gene which can be a significant biomarker for the TB resistance, especially the C allele at 869 and the T genotype at 509, where they are trying to provide a substantial protection against the developing active TB and the experiencing a relapse in the patients. Okay. So this is another group where I have also studied with the recombinant cytokines, whatever the cytokines which we are able to see, they are in the study. So further we have extended our study to to study with the recombinant cytokines. So we have studied the recombinant cytokines of both the pro and the anti-inflammatory cytokines. We have taken the pro TNF alpha and the IFN gamma and the anti-TGF beta and the IL-10, and we were trying to look both in the TB patients and also in the household contacts. So in the latent TB is nothing but in the household contacts, and in the general population also we are trying to we are expecting and we are experiencing also. So the latent TB, it helps in that recombinant TNF alpha. In the latent TB, it helps to control the infection by promoting granuloma formation where it contains the bacteria. And active TB, the excessive TNF alpha contributes towards the chronic inflammation and tissue damage where it tries to worsen the disease progression. And IFN gamma, if you see recombinant, the latent, it will activate the macrophages to kill the MTB and also keep the infection under control. Whereas the active TB, it may be less effective during high bacterial load, but it contributes towards the chronic inflammation and the disease severity. And TGF beta, recombinant TGF beta, latent TB, it regulates inflammation and supports granuloma formation, where it tries to help in controlling the infection. And in case of active TB, it reduces the immune response and promotes the fibrosis, where it trying to contribute towards the tissue damage and impairment which is trying to heal. And the last is the recombinant IL-10. Here in latent TB, it regulates the inflammation and prevents the excessive immune response, aiding in the infection control. And the active TB, it suppresses the immune responses, allowing the bacteria to proliferate and worsen the disease. So these are the important, like how they are going to function, both the pro and anti-inflammatory cytokines. So here we have done the lymphocyte proliferation with the MTB antigens, both the ESAT6 and antigen 85A in both the patients and the household contacts. So both the antigen 85A and ESAT6, they have shown a low lymphoproliferative responses both at the 0 month compared to that of the HSC and the HC. But we have seen a significant difference was observed only in the ESAT6 between the 0 month and the 6 months during the follow-up. And we have also stimulated with the recombinant pro-inflammatory cytokines, both the TNF alpha and the IFN gamma, and we have observed that a low lymphoproliferative responses were observed in APTB patients at 0 month compared to HSC and the healthy controls. And also with the anti-inflammatory cytokines, that is a TGF beta and the IL-10, both have shown slightly lower proliferative responses in APTB at the 0 month compared to that of the HSC and SC after 6 months, but there is no significance at all in these anti-inflammatory recombinant cytokines. So what are the key findings of this is, if you stimulate with the ESAT6, we have seen after 6 months a highly significance. And the pro-inflammatory cytokines, they also were highly significant, but the anti-inflammatory, that is the regulatory cytokines, were not to be significant. So what we conclude from this is, maybe the active TB induces a state of selective immune suppression after antigen specific and pro-inflammatory pathways. So if you see the successful treatment over 6 months, maybe they effectively restore their proliferative responses, bringing the patient's cellular immune profile back towards the baseline observed in the healthy controls and also the people who are being exposed with the TB. Okay, and this has been published in Cytokine in 2024, this data. And another part of this is a apoptotic marker, that is a MCP1. This is a monocyte chemoattractant protein which functions as a double-edged sword in cell survival, with its impact on apoptosis, which will dictate by the specific cellular environment. So these are the dual role of these MCP in the apoptosis. I am not going to go into the depth. So here we have stimulated the MCP, like we have stimulated with the antigen 85 and ESAT6, the PBMCs, and we have in the supernatants, we have estimated the chemoattractant protein, that is a MCP1, and we have observed 0 to 6 months both, like we have not only stimulated with the antigens, we have also stimulated with the pro-inflammatory and the anti-inflammatory recombinant cytokines, and we have estimated the MCP levels. And here we are able to see that the MCP levels were significant during the follow-up. So with antigen, the higher MCP in healthy controls and contacts was observed when compared with the active TB, and the pro-inflammatory cytokines, they were high in the household contacts compared to that of the other groups, and the anti-inflammatory cytokines were high in the active TB patients compared to the only for the anti-inflammatory, that is a recombinant IL-10. So what we can conclude from this study is the household contacts exhibit the most robust MCP1 responses to pro-inflammatory stimulants, but whereas the healthy controls and contacts together show higher MCP levels in response to MTB antigens compared to that of the active TB. So these patterns serves as one of the critical indicator of the disease state and the immune challenges in these subjects. And this has also been published in the Cytokine in 2024. And last, it is throughout my study, it has been what we have observed is the active pulmonary TB patients who are turning into relapse, that genotypes we were just trying to look into them, and we have observed that 2.5% of the active TB patients turn to relapse within 6 to 12 months of the treatment. They have shown susceptibility towards the genotypes of IFN gamma, IL-10 and the IL-12. And even the contacts who are developing the disease, we were saying the contacts of the TB patients, it is a study which I on my own I have done in a DOT center at Mahavir Hospital, where I have observed that the contacts of the TB patients, they are developing a disease within 6 months to 2 years of their time period. They are coming back as a patients. So we have observed their genotypes also. So we have seen that these are the different cytokines, IFN gamma, IL-12, 2, 4, 10 and TGF-beta, these are all they were showing the susceptible genotypes. So we feel what, screening of at least 2 susceptible genotypes may help us for identification of the high risk individuals, not only in the contacts, even in the general population. So this is our observation, and this is one of my contribution towards the nature immunology with the Indo-German collaboration, where part of my research work has been contributed to the nature immunology publication in collaboration with the University of the Berlin, Germany. And currently I have 2 ongoing projects like DST Sure, and another one is a DBT builder. This is a university grant, and so far I have completed 15 projects which have been funded by various funding agencies like DST, DBT, ICMR, DHR, COE, DBT, Cadila. I have done 2 clinical trials, that is with Cadila Pharma and the Serum Institute of India, which is in collaboration with the Report India. And also there was a big project where Indo-US TB VAMP, which has been jointly funded by DBT, ICMR and the NIH. And these are my students. I would like to thank them, because without them I would have not generated so much of data to present today here. Thank you.
Thank you so much, madam, for such a wonderful, clear, informative exposure on tuberculosis disease. On to the short tea break of 10 minutes. We'll be back in 10 minutes again. I request audience not to carry any eatables into the auditorium, please. Thank you. And speaker of today's event, Dr. Anuradha Udimudi Garu. Over to you, Kadaveri.
Anuradha Udimudi is founder and chief scientific officer, GeneTech, Hyderabad. Dr. Anuradha earned her PhD in human genetics from Osmania University, Hyderabad for her groundbreaking research on risk assessment in cervical dysplasia, a precancerous condition of cervical cancer. Her research was honored with the Best Research Presentation Award at the Environmental Mutagenesis Society's 26th annual meeting in St. Louis, USA. Dr. Anuradha is one of India's most influential medical geneticists whose contribution to bringing cutting-edge advances in genetics and genomics to patient care is well recognized by the physician community. Her transformative contribution to medical practice continues today with her latest innovations in the field of fertility genomics. In her 30+ years of serving as country's foremost medical geneticist, Dr. Anuradha has given more than 600 lectures in international, national, state and local conferences and CME programs, and is recognized as a key opinion leader technology companies of the world. In 1998, Dr. Anuradha established GeneTech as India's first independent medical genetics center that offered comprehensive genetic testing with counseling. Under her leadership, GeneTech continues to produce groundbreaking innovations in the field of reproductive medicine and fertility medicine. Through grit and grind, she carved a niche for herself and comes across as an epitome of poise and grace. It's our honor to welcome Dr. Anuradha Udimudi to deliver her address.
And I really love to be around young people. So this is one of the opportunities Dr. Padma and the principal of Aurora have given me. So I'm very happy about it. I hope I will share some of my experiences. Hello? Yeah, which will help you guys to see something through my eyes, the way I have experienced this field and this profession. Can I have the slide, please? Sure, no problem. So meanwhile, I'll tell you probably a little bit about myself. I'm a very, very average person. You can see, as you can see, maybe not average, but even shorter. But I never was first in class. I never scored more than 95 any time in my life in any subject. I was a very, very average person, maybe above average, a first class student. But because we did not have cell phones and we did not have too much TV time, I was allowed to dream. And so those dreams, you know, a passive dreamer, that you want to become something, made me what I am today. And I think the generation today is missing that dreaming. And because you are just dreaming what is being shown to you, but there is no originality in your dreams because you don't have free time. And I feel that that kind of dreaming, that kind of, we are not doing anything, just sitting around and thinking about something, is very, very important for human beings. That's what makes us humans. And that is where your passion develops. And during my childhood, I wanted to become a doctor, like many of you here. How many of you wrote MSET wanting to become a doctor? I'm sure a lot of them, right? So when I did not become a doctor, I wanted to be still in this field, the medical field. No, my slides don't want to come today. Should I move? So, and that is how I have developed a passion. Now, my passion is towards medical system. I wanted to be a doctor, I wanted to treat patients, I was interested in health aspects, and therefore I pursued to do something in the area of medicine. And even in the area, I have had several opportunities. Then, you know, I've done my PhD. During my PhD also, luckily, I was in the field where of cancer, where I was working on cervical cancer patients, and I was trying to pick up in the predisposition state. I mean, you see, we have what is called a predysplasia and then cancerous condition. So in the predysplasia condition, which is like a pre-cancerous condition, I was trying to pick up which are the individuals which will go towards cancer. And this we have done using DNA damage and repair mechanisms. And we have found that people with higher DNA damage and poor repair capacity are the people who will go towards cancer. And this was my work. The same work was also supported by my studies, which I have done in CCMB, sir. Dr. Thangaraj sir is here, and we are very honored to talk in front of him today. And CCMB work that I have done also proved that the mice in the precancerous condition with higher DNA damage and low repair capacity can or have developed tumor in future. So with that, any luck, sir? Okay. So this is fine. So then after this, just I'm telling my story, which is a little bit boring probably, but it's okay. You can get something out of it. So once I finished my PhD, I moved to US for a postdoctoral fellowship, and I worked in the area of genetic toxicology. Genetic toxicology work that I did in a private company was on cell phones. Cell phones were coming at that time. The markets were being flooded, and the cell phones looked this big at that time. And those cell phones, some people have, said that they cause brain damage. So my work was to find out if they really cause any brain damage. Luckily, I found out that they do not. So you all are safe now. And we did extensive work on cell lines and mice, and we found that the radiation does not cause any brain tumors. This was actually a case that happened in US, which was later on funded. The government has funded several research projects. One of the research project was mine, and I have demonstrated that it does not. I also worked on something which a paper that is being quoted in many of the space research papers today, where I have shown that freezing cells for a longer time, does it cause DNA damage or not? And again, it's been proven that it does not cause DNA damage to the core. And these are the papers that I have worked during my postdoctoral fellow, but I was never satisfied because I was in the lab doing work, doing wet lab work. I'm a poor wet lab person. I'm a very clumsy person. I drop at least one glass every day. And you can imagine how I'll work in the laboratory. I wasn't very, very happy in my wet laboratory. And I wanted to go back to medical field. And that's when in US I moved to a laboratory called Laboratory Corporation of America. It was previously called as Roche Labs. There I have joined the cytogenetics department. So in the cytogenetics department, you know what cytogenetics is all about, right? It's about looking at the chromosomes. So I worked in cytogenetics department for some time, and then my very, very strong passion of coming back to India and establishing a lab of my own, because I know that at that time there were no labs in the country which were offering good medical genetics services, genetic diagnostic services. I wanted to be in the field of genetic diagnosis. So I also got some experience in molecular biology. Oh, thank God. And then I came back to India and established GeneTech in 1998, long time back. So yeah, we'll now move on. I'll just show you some slides in the second slide, sir. Okay. Now, what am I doing? I don't like these podiums because most of the time I am not visible. And that's why I like a hand mic. And this is the problem that I am addressing every day. Every day. What am I doing at GeneTech as the founder and head of GeneTech is I am addressing this question of doctors. Doctors look at a problem. 50% of the time, they're able to solve the problem. 50% of the time, they don't know what they're looking at. Believe me, 50% of the time. And that is when they are thinking, could it be genetics? Maybe it is genetics. That's the answer they give. But who will find out whether it is genetics, and if so, what needs to be done? Next slide, please. Next slide, sir. Thank you. So these are all the fields where we contribute as a genetic laboratory. You can see that the first row is completely filled of reproductive medicine. So we contribute and we find out what is the cause for infertility. And gone are the days where genetics was, correlated only with chromosomes or cytogenetics. Now we have so much more to offer. We sequence human genome today in the laboratories, and we do it in bulk, pooled up samples. I will tell you a little bit about that. And we identify what's the cause for infertility, and we also provide information where we tell doctors how to treat infertility. So personalized treatment, personalized precision medicine is already here. Personalized medicine meaning that we all have headache in this room. Of course, many of you already must be having now. If you all take the same medicine, some of you will have relief, some of you will not have relief, some of you may develop some other side effects. And why is that? Because each one of us are very, very different. And why are we very different? Because our genes are different. Our susceptibility is different. Therefore, treating a person based on their these differences is precision medicine. And that happens at the infertility level also. How to stimulate your eggs, which drug should be given so that you get more eggs. How do we ensure that your sperm count increases? What drug will help you increase your sperm count? All this is dependent on genetics, which we do. Okay, and then pregnancy. Pregnancy is so many issues, pregnancy losses, pregnancy with an abnormality. So we diagnose all these conditions in the laboratory. Newborn. When a newborn is born, a child, there are so many issues that may come up which may not have any answer. Again, the doctor is thinking what is happening. At this time also genetic helps in diagnosis, and also during the development. You can also see that there are a lot of children with autism these days. All these conditions of mental development and physical development are being diagnosed in genetic laboratories. Then cancer is one of the very, very major application where based on your genetic change, your cancer will be treated. So cancer, endocrinology where your endocrine functions and even the development of including female development, all those are endocrinologist's area which also has genetics impact. Cardiology, coronary artery disease, blocks in the arteries, heart attacks, all these are because of genetics. Psychiatry, n number of neurodegenerative and neuromuscular disorders are because of genetics. Blood related disorders are because of genetics. You can name some: beta thalassemia, hemophilia, and many others. Ophthalmology, you name night blindness, retinoblastoma, all these are because of genetic underlying factor. And there are other applications also, like organ donation. If there is an organ donation, one must know whether there is compatibility. And this compatibility is also studied using genetics. By looking at the genetics genes, we will identify whether the organ compatibility is good or not. And then pharmacogenomics. Just like I told you about the headache, there is pharmacogenomic application in cardiology. So whether to use beta blockers, whether to use some other drug to help you relieve your hypertension or BP is based on this area which is called pharmacogenomics. And then infections. Infections, also your response to infection is also based on genetics, and this is being used. And nowadays geriatric care for older people is also one of the things that we are doing regularly. So the technology is going fast, and you guys have to catch up. And more than you guys, I think the government and the education system has to catch up. And because the reason I am telling this is when I interview people, there's a huge gap of what's happening out there and what these kids are coming out from our universities. And so I don't know what to do, but something has to be done. As a group and as elders sitting here who have power, I think we have to do something to bridge that gap so that people catch up. And of course, you all know what chromosomes are. Chromosomes have the DNA, which has the genetic code, which produces the protein. So we can study a genetic disorder at the level of chromosome, at the level of DNA, or at the level of protein. Next slide, please. So, but there is a lot of difference. When I say then, I mean 1998 when I started GeneTech. And when I say now, I mean now. And the technology has improved so much. For example, they just put up 2 examples where if we were actually, some of you, my colleagues will remember, we were taking pictures of chromosomes, cutting them, pasting them, and it really was frustrating. And now we have image analysis which does this 15 times faster. You just put the slide under the microscope, it captures by itself, it separates the chromosomes by itself, it just gives you the karyotype, you just have to look at it and then make small changes, and that's it. So it's now 15 times faster. And what we used to do in the PCR laboratories, prepare the PCR, load it, see the gel, see where the band has come. If the band has not come, why did it? It was so messy. But now what we do is sequencing. And when we sequence, we can actually see the complete genetic code of the area that we have sequenced. So we can sequence single genes, we can sequence multiple genes. We do what is called a whole exome sequencing. You all know what is the difference between an exome and an exon and an intron, right? The gene has an expressing part which is exon and a non-expressing part called intron. So if you combine the expressing parts of all genes, it's called exome. So we do exome sequencing of 25,000 genes today by a whole exome sequencing test, and we have an ability to pool cells. I can actually pool 25, 50, 100 samples, all DNA samples in one tube, and then I can sequence them. And I know that this is Dr. Sumanlatha's and this is Dr. Thangaraj's and this is Dr. Padma's DNA. Because there are barcodes that we add to these DNA which tag to them and then identify. So this technology has improved so much, and even the cost, we used to have around thousands of dollars for one gene. Today, the cost of sequencing 25,000 genes is not more than $200. So, and it's becoming cheaper and cheaper every day. So that is where the technology is going. Next slide, please. But then, that is Dr. Anu sitting in the laboratory. It's not easy. It is definitely not easy when you are doing diagnosis. When you are doing research, I'm not saying that research work is easy, but especially when you are going to do diagnosis, the result is going to come right then. In the sense, if I give that this baby in your womb is going to be normal, and within next few months the baby will be delivered. And if that baby is abnormal, it's over, right? So you have to have extreme amount of quality assurance processes in place in a laboratory which is diagnostic. And then we have to look into all these things. Now, as I have told you, previous slide please, as I have told you previously, no, next slide only. Next slide, next slide. Yeah, that slide. Same slide. So you can see here, from each patient we are generating 10 GB of data, 25,000 genes and 40 million base pairs. 40 million databases, of which we are again picking up around 20,000 variants. So for each patient, if I have to check 20,000 variants and report something that is relevant to that patient, you can imagine what's happening in the lab. Of course, there are a lot of software, pipelines, bioinformatic tools that are available. Still, you will need a lot of expertise time into this. So, and sometimes the variants that we get are called variants of uncertain significance, meaning we really do not know if they are disease causing or not disease causing. In such a situation, what do? So we have to report. According to our guidelines, it says it is an uncertain significance if there is no publication of this variant in the literature. But the variant is correlating with the phenotype. Phenotype is the presentation of the patient. Then we are in a dilemma. So we think a lot about it. So there are a lot of complexities that are involved in reporting, and this is called as data analysis. I'll just give you an incidental finding example. For example, patients have come to us saying that we both are related to each other and we have a child with retinitis pigmentosa and we don't want another child with retinitis pigmentosa. Okay. Now, so we did sequencing of the couple and we found that they are carriers of retinitis pigmentosa. Okay. So far you all are with me? They are carriers of retinitis pigmentosa. But I also found, because we are seeing everything, whether I want to see or not, the sequence is showing there. We are seeing everything, all the data of that individual. And I have found that the female partner is a carrier of breast cancer risk gene variant. And male partner is a carrier of late onset disease, Alzheimer's. Now, tell me if I have to inform this to the patients or not. How many of you think they came to me to find out if they have a risk for another child with a genetic problem? That was their intention, primary intention. But incidentally, I found something else. How many of you think I should tell them? Huh. And how many of you think I should not tell them? Okay. So, majority is saying I should tell them. And so the same debate happened in scientific community also. And we have debated quite a bit internationally. And some people said, it is not your DNA, it is their DNA. Who are you to hold back that information? If it is their DNA, you should tell them everything, whatever they have. Some people on the other hand said, if you are telling a person that you are going to get breast cancer by the age of 65, and she is like 26 years old, you are destroying their lives, you know, for the next 20, 30 years, whether she may or she may not develop. It's a risk that you are going to give, right? So then the debates happened for a decade actually, and then we got guidelines saying that, okay, let's compromise. Let us report only if a patient wants to know. So all laboratories now take a consent saying, we have secondary findings or incidental findings that we may see. Do you want us to report or not? One. Second, the labs only report a list of genes for which there is a management available. So if you have a breast cancer risk, there is management available. In the sense you can go for mammogram every year or once in 2 years, and you can self-examine your breast. You can do a lot of things to prevent breast cancer. You can be on oral contraceptives, tamoxifen is a good option. So all these options are available to prevent breast cancer. So therefore BRCA gene is involved in the list. If there is nothing available and it cannot be resolved, then risk cannot be reported. So there are a list of genes that we have from American College of Medical Genetics, which we use for reporting. Next slide, please. So I just thought I'll just tell you what is the sense of accomplishment for a geneticist from my perspective. The journey that we took, when I say we, it's my family also, who took this journey along with me, and some of the very seniors who are still with me from past 15, 20 years, and the challenges that we have faced. So I always think that you don't just wake up and become a butterfly. You have to go through all that caterpillar pupa thing and all. The challenges were many. You will not believe, when I established in 1998, what I did, I had my very old Premier Padmini car that of my father-in-law's, and I used to drive, that was a stick shift car, and go to each and every doctor to tell them and ask them that I want to establish a genetic laboratory. What do you think about it? You will not believe, 99% of doctors said, are you crazy? Why do you want to start a genetic laboratory? What can you do with it? There is no treatment for genetic disorders, right? So what will you do by diagnosis? I already know this baby is a Down syndrome. By looking at the baby, I know it has Down syndrome, the features. Why will I use diagnosis? What help will I do? So the idea of prevention, that we can prevent birth of another child in the family with genetic abnormality, did not exist in 1998. Believe it or not, I visited so many doctors, told them, and very few of them, that 1% of them said, it's very good. There is no private organization which is doing this. You should do it. So then the whole thing started. I took a small duplex flat and established a laboratory. I was my first technician in the lab, person who did cultures, looked at purchased, my mother was a bank manager, so I managed to get a bank loan. So 25 lakhs is the loan the GeneTech started with. We put in some money. We didn't earn much in US because whatever reasons. And we came back, both me and my husband. And my husband started a software company and I started GeneTech. And we took a small duplex flat and we started working in it. And my first employee, I hired by name Satish. and then Kumar, I think. So these people are now directors in big pharma companies in US. And they were my first employees. They joined after MSc. They helped me set up the laboratory. And you know what? We had a big party on 31st of a month when we got 10 samples. The day we reached 10 samples, we had a big party. That's the kind of volume, that the kind of business that we had when we started GeneTech. Today we do thousands of samples, but then it is a journey. It requires perseverance. There were many days where I thought it is impossible. It is impossible to do what I saw in US. What I visualized that a laboratory will do in US, I am sure I cannot do that in India. But no, I stuck to it. Then we continued to do it. I became pregnant. I had my child, first child, both girls I have. First girl was born soon after I started GeneTech. 14th day, I took her to GeneTech. Every day I used to take my daughter to GeneTech. I used to feed her there. My employees used to, she grew up with them and she crawled there, she walked there. So it's like home. My lab is like a home for my child. And then the second one came. And then because I understood that it is very difficult for me to grow if there is no awareness, every weekend, whether you believe it or not, every weekend, both me and my husband used to take a car and go to remote districts. Warangal, Khammam, Vijayawada, Nalgonda, you name it. Every single doctor in Telugu states would know me, at least the gynecists and obstetricians and infertility specialists and pediatricians. Because we would go, organize a camp, organize a lecture, I would speak with the doctors, develop rapport, give them. Every week I used to prepare a case and share with the doctors. So there's a lot of work that has gone into it. And today I'm really proud that we are the most trusted genetic laboratories in Hyderabad, serving both Telugu states and also getting samples from all over the country. Next slide, please. Next slide. So this is one I just thought I will show you some use or applications of genetics in laboratory, especially in reproductive medicine, because that's where I am very, very strong. And if you see that close relative marriages, they have high risk for genetic disorders. And you see that Telugu states and South India is in red. So you have lot of consanguinity, close relative marriages in South India, which will give rise to much more genetic disorders. And therefore, you can actually, this picture is very clear. It shows that unrelated people don't share mutations, but related people share more number of mutations. Next slide, please. Yeah. And then once you know there is a risk, what do? You can't remove the mutation from that person. Of course, there is gene editing and gene therapy, which is yet to come in a mainstream practice. So all you can do is prevent. And how do we prevent? We prevent by doing testing of the fetus, and now we do what is called testing of the embryos. So how do we test fetus? We test once they get pregnant. Right? Let's say somebody has a risk for some genetic disorder. So when they get pregnant, they come to us, and during the first 3 to 4 months, we take fluid from their womb or a biopsy from the fetus. We test and tell them whether the baby is normal or not. If the baby is normal, they will continue the pregnancy. If the baby is abnormal, they will terminate the pregnancy. And terminating the pregnancy goes against a lot of their beliefs. And also emotionally, it is not very, very easy for the mother and also for the father. In such a scenario, we can use what is called a embryo testing. That means you are not actually, you are making the embryo outside, in vitro fertilization. You take sperm, you take egg, you make the embryo, and then you do a small biopsy of the outer layer of the embryo, and the cells are sent to the laboratory. We retrieve the DNA and we look for the mutation, and we say, yes, this embryo has mutation, this embryo does not have the mutation. whichever embryo is normal, they will transfer that embryo into the womb. Now that will increase the chances of having a, I mean, of course it's a normal baby, but at least you don't have to go through the termination of pregnancy, which is a very painful situation. So these are some of the applications that we do in reproductive medicine, which gives me extreme amount of satisfaction that patients come back with sweets saying that, yes, I have delivered a baby, you have helped us in conceiving with infertility, and then the baby is normal, and things like that. Of course, we'll also have to face diagnosis where sometimes we give abnormal reports, and that requires huge amount of counseling. I have not gone into details of gene therapy just because we are not doing it. In our laboratory, it is not a practice, but these are some of the disorders for which gene therapy is already in place. However, in India, we have a very rare access, and some are being facilitated. It's very, very expensive. As example, SMA has a possibility of getting gene therapy done, but it's very expensive and needs crowdfunding. And most of our trials are in very, very early stages, and I'm sorry to say, but there is no indigenous manufacturing mechanism here. And most of the gene therapy that we today have, we import from abroad. And I'm sure at least one of you in this room will go forward and set up something in gene therapy area in the drug industry in India so that we can produce indigenous therapies in India. Next slide, please. And what is most important thing that we do in our lab is genetic counseling, and communication is everything. And COVID has taught us quite a bit, actually, because people used to come from Gulbarga, Belgaum, some places, travel all the way through car, being pregnant, etc. But then now COVID has started that you can do everything online. Everything, shopping, counseling, everything can be online. So this is what we are doing. And today, most of our genetic counselors, who are all PhDs with at least 10 years of experience in the field, speak with patients online. And some of them, of course, they do come and speak to us. And we did around 1 lakh genetic counselings in GeneTech so far. And what does genetic counseling involve? It involves looking at medical reports. It also involves physical examination of the babies. We have to take 3 generations of pedigree, and then we extract data and we use genetic databases to identify what could be the genetic condition. We discuss prognosis and recurrence risk if the report is already available, and we also write a letter to the patient. Here I just want to tell you a small story. I'm come almost to the end. This was again in the very, very beginning of GeneTech, where I did not even have this duplex small laboratory, flat laboratory. I actually started counseling patients in my home. So I cleared the hall. There's a dining table. So on that dining table, we would actually, patients would come, I talk to them. Later on, the duplex flat started, and then a small lab established later on. In those times, early times, a couple came with a child with thalassemia. And thalassemia, as many of you would know, is a recessive disorder. That means both the patients, both the couple, husband and wife, are carriers, having out of those 2 genes, one gene having a mutation, the other being normal. So what are the chances that these 2 come together? Is 25%. That means mother can give mutation, father can give mutation, 25%. The other 75% is mother can give mutation, father need not give, or both of them need not give, right? 25%. So when I explained this with the help of illustrations and all that, and I told them, look, you have 1 in 4 risk of having a child with thalassemia in next pregnancy. So you have to come back to me when you get pregnant. So we would do testing of the fetus using amniocentesis, taking amniotic fluid, because at that time embryo testing was not available. Now, they went back. After 2 years, a doctor calls me and she starts screaming at the top of her voice and saying that, how could you do this? They have delivered another child with thalassemia. It seems you told them that there is no risk. Now, obviously, even in my sleep, I wouldn't say that. I am breathing genetics. And so there's no way I can do that. I said, no, no, ma'am, you please send them back to me. So the patients came back to me. And then I said, I've told you there is 1 in 4 risk, right? मैंने आपको बोला है 1 in 4 risk है. So they said, हाँ मैम, आपने बोला है 1 in 4, एक तो abnormal हो गया तो बाकी 3 तो हम normal सोच रहे थे. For people who can't follow Hindi, they are saying, అవును మేడం మీరు చెప్పారు ఒకటికి 4 ఒక రిస్క్ అన్నారు కదా, ఒక రిస్క్ ఆల్రెడీ బేబీ పుట్టేసింది, మిగిలిన 3 నార్మల్ అనుకున్నాను. So what do you think is problem here? I mean, what happened? It's a costly mistake, right? What happened? What happened in this situation? Communication failed. Here, communication failed. I should have said 1 in 4 in every pregnancy. 25% का मतलब ये नहीं कि first 25% हो गया तो next 75 is going to be. It means 25% risk in every maybe I did not use the word in every pregnancy, or maybe I used it, but patients want to hear what they want to hear, okay? This is another thing I learned. You say 100 things, they will only hear what they want to hear. So they must have gone and told the doctor that madam ne no risk bol diya hai, 1 in 4 1 ho gaya hai, the other 3 are going to be normal. So then I realized that this is not the way genetic counseling has to be done. So from then onwards, we do not let the patient step out of our center until we give them a written letter of recommendation. Get a signature and keep. So this is very, very important. You have to communicate in proper language. And the language of genetics, communicating genetics to patients is an art. Communicating genetics to doctors is a different art. So this must be, this is very, very important. Knowing facts is not at all important. Knowing 25% risk is not important. How do you communicate that to the patient? What illustrations are you going to use? How patient are you to listen to them? I mean, first you ask them a question, do you have a family history of any genetic disorder? They will say, no, 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 no. Then you ask, how many siblings do you have? I have 3 sisters, madam. All of them married? Yes, married. Okay, how many pregnancies did they have? My first sister had 3 pregnancies, 2 she lost and the last one is now fine. Okay, so the 2 pregnancy loss information has come now. From the first to no, right? So this kind of interviewing, having patience and not being judgmental. I mean, sometimes we have families, I don't do counseling anymore, but I used to enjoy it a lot. When they come with husband side family, wife side family. The husband side family, మా ఇంట్లో ఏం లేదు మేడం. It must be their own. మాకేమున్నాయి, మాకు అస్సలే లేవు. So it's like you're also doing a psychological thing and the couple are sitting quiet, not talking. So then you, at some point you will have to say, maybe the extended family has to step back. You will have to talk to the parents, couple. Even couple have problems. So even that, but you cannot be judgmental. You need facts, but you will have to talk to them in a way. So this communication is an extremely important thing in genetic counseling, and that is why, because I don't hire anybody who is not trained under me. Yeah, it's weird, but that's what it is. So under me, if they are trained for 5 years, 3 years, I'm then comfortable that now you can go and talk to patients on your own. So all my genetic counselors, PhDs are very, very good. And this is one field. Next slide, please. And during this process, I have also created an IP. It's like I felt like a scientist. I worked like a scientist, and when the sequencing technology has come, we have used around 5,000 samples of infertility patients that I have previously had, and we have created an IP, intellectual property, for ourselves, and created 2 analysis tools called GeneAndro for male and GeneAndro for female. And these tools actually help infertility doctors today to do personalized medicine and to give personalized treatment to infertility specialists. They identify the root cause, they give drug implications, they also tell what protocol to be used, how much drug should be used on this woman to get good eggs, how much drug should be used on this man to get better sperms. And then they also talk about donor and surrogacy options. So donor options cannot be, if you have two IVF cycles failed, generally the guidance is go for a donor egg or go for a donor sperm. It should not be like that. It has to be evidence based. And some of the genetic information that comes from these will tell the doctor that this is the patient, you have to use a donor gamete. So these are some of the things that I have done, and a very, this is the sense of accomplishment again. Next slide. And then there is this area of wellness genetics that I thought I should clear some doubts here. Wellness genetics is very different from medical genetics. Medical genetics, clinical genetics is mostly identifying genetic disorders in the family, where we test the mutation and we tell whether the baby has the genetic disorder or not, and other things, pharmacogenomics, etc. But here in wellness genetics, what we do is risk prediction. That means everybody these days, I think the young people now, Gen Z, they want to do a lot of fitness and good. I mean, we were not like that, we were dreamers, but good that you want to do this. And therefore they have questions. Oh, am I prone for diabetes? Am I prone for heart disease? Do I have vitamin C deficiency? Will I get vitamin D deficiency? All of us have vitamin D deficiency, okay, by the way, in India. So don't worry. So these things, and if you know that you are at a risk for certain condition, you can take preventive management. And this is wellness. Wellness is not when you have a disease. When you want to prevent a disease, you want to understand your genetic profile, correlate it with your other medical reports, and take preventive precautions so that you will not develop the disease is wellness. This is a consumer genetics it's called, and this is used for many things. For example, even in hair transplant, we get a lot of cases where whether this drug should be used for hair transplant or that drug should be used. Obesity, junk food. I eat junk food, I put on weight. My husband eats junk food, he will not put on weight. Now, what is that? Because we are different. We have different genetic compositions. And so, because I have a risk, my carbohydrate should be lowered. Fitness, some people are runners, some people, even in sports, people are using this, where they want to see if they can make a career as a runner or, as a swimmer, etc. And what kind of nutrition, how to control your cholesterol, even addiction to alcohol, addiction to smoking can also be genetic in nature. And these reports are really fun. So you don't take them very, very seriously, but they do add a good amount of information to your risk. And for people who are interested in well-being, this is an excellent opportunity to know their risks. Next slide, please. So this is almost my last slide. I put up whatever came to my mind. I just put up what all careers that you all can choose in genetics. You can become a medical geneticist or a clinical geneticist. You can become a molecular genetic technologist. You want to work in the wet laboratory, you can do that. You can become a cytogeneticist. You can become a biochemical geneticist. You can become a genetic counselor if you have good communication skills and things like that. You can become a bioinformatician. So there is lot of data that is being generated from these sequencing machines. So we have to develop all kinds of pipelines to analyze them. So you can become a bioinformatician and you can become an AI related expert. You can become a biostatistician in genomics. There is lot of requirement for although there is AI these days, but you can become a biostatistician. You can become a data analysis expert. You can become a forensics expert. You can become a researcher like Dr. Sumanlatha Ma'am. She worked so much on IL genes. You can become a researcher. You can become an expert in gene editing. You can go in drug industry, you can do pharmacogenomics, you can go to biotech industry, plant and animal genetics, which I have not covered in my lecture, does also has a big scope. You can become a quality assurance expert. You can actually become, the lead auditor. You can become a professor in genetics. You can become an entrepreneur like me. You can become a lab director. You can become a manager. You can also become a business development person if you are joining an equipment company or a drug industry. So the options are there. I really don't buy the thing that there are no opportunities. There are opportunities. You just have to look for them and you just have to be focused. Next slide, please. And my piece of advice to youngsters is make passion your profession. If you can do that, I think you will be happy. And if you are happy, you will do good things. And please dedicate at least few minutes, so much time you all are on cell phones. Give 15 minutes to genetics if you want to build your career in genetics. Every day give 15 minutes of browsing on genetics. You will find something new, something might interest you, something will lead you somewhere. So I think that is important. And please publish. I have published 8 papers. I don't want to reveal my age, but yeah, 8 papers at this age. And in last 2 years, just in last 2 years. So youngsters have to write. Our education system is not letting our people, our students build communication and writing. But you have to write and start publishing today. It's okay even if you publish in a small journal. It doesn't matter, local journal, local magazine, doesn't matter. Write and publish. This will help you in your career. Perseverance. Don't be disappointed if you don't get something. Something better is in store for you. You work towards what interests you. And then always aim for higher studies. I always tell everyone, whatever is best possible, you do that. PhD is the top one. Get a PhD as much as possible. International exposure is very, very good. If you have an opportunity to get international exposure in this field, you should do it. Work experience in good labs is an added advantage. And have fun. Have fun while building your career. Enjoy. Enjoy your time, enjoy your family, do your hobbies, and still love your work and write. And with that, I think small advice, I want to wrap up, and I will look forward to question and answer sessions, and so we can interact more. Thank you, one and all.
Thank you so much, madam, for an enriching disclosure on medical genetics. Madam, you gave a small dive into the genome field and enhanced our knowledge. Thank you so much, ma'am. Now, I take this honor to introduce our keynote speaker of today, Dr. Kumaraswamy Thangaraj. Dr. Kumaraswamy Thangaraj was born in Chennai, Kanchipuram, Tamil Nadu, and completed his BSc, MSc, MPhil, PhD from University of Madras. Dr. Kumaraswamy Thangaraj is a CSIR Swarup Bhatnagar Fellow, CCMB Hyderabad. His major research interest sits in the fields of human origin, health, diseases, especially in molecular basis of cardiovascular diseases, infertility, sex determination, mitochondrial disorders, genomics, forensic genetics, and he is also interested in understanding the impact of endogamy on health diseases of Southern Asia. Dr. Kumaraswamy Thangaraj has published widely in such peer-reviewed journals as Nature, the American Journal of Human Genetics, Molecular Biology and Evolution, and Current Biology. Dr. Thangaraj has been bestowed with numerous awards and fellowships, including United Kingdom India Education and Research Initiative, awarded by Gordon Brown, British Prime Minister in 2011, Raman Research Fellow Award in 2007, Rashtriya Ratan Award in 2006, CSIR Swarup Bhatnagar Fellow, and former Director of CDFD Hyderabad, and more recently was conferred with Padma Shri by the Government of India. dedicated 3 decades of research in understanding the complex origin and affinities of Indian population using the genomic variations of contemporary population and ancient biological remains, and has driven the National Genome Initiative. One of the most celebrated achievements was the discovery of that tribal population of Andaman Islands are the direct descendants of the first modern human to migrate of Africa and southern coastal route approximately 65 years ago. People who are conversant with Tamil may know Thangaraj sir's name, Thangaraj, the king of gold. Gold rally may show fluctuations due to the global uncertainty, but Thangaraj sir's achievements and persona will always show a J-shaped growth curve exponentially. A giant pioneer in his field, Dr. Thangaraj Garu is modesty and humility personified. Any words of praise is like holding a candle to the sun. Whole student and teacher community is indebted to sir for his benign presence here today to motivate and spur us on our scientific journey. We are all eager to savor a few drops from his ocean of knowledge, and we are pretty sure that his talk today will lead to an unquenchable thirst of more knowledge and catalyze and drive our own scientific progress and journey. Welcome Dr. Kumaraswamy Thangaraj Garu to please bless with his knowledge and wisdom. We welcome you, sir.
To all of you. So after two excellent talks, so I am going to talk to you something different. I hope you all will like this. So mostly how human have originated, how our forefathers have reached India and spread across the world, and how our culture, I'm not talking about a lot of cultures, I'm talking about only endogamy and consanguinity, which Dr. Anuradha also highlighted, as impact on our health and diseases, right? So to talk about human origin, you all must have studied, right? We have originated from a non-human primate. Right? If you look at this figure, the human and chimpanzee have originated from a common ancestor called hominids. Okay? And the reason why we are saying these two is that we share about 98% of our genome with chimpanzee. Okay? And that 2% of variations which are present in a several genes are the one makes us as a human. So I'll give you one example. Just take one particular gene, human accelerated region. This gene is essential for our brain development. Okay. If you see how the mutations are accumulating in our genome, particularly the mutation which are functionally relevant, from chicken and chimpanzee, there are only 2 mutations. The gene is the same, and the mutation, those genes are essential for making the difference. Only 2 here, but between chimpanzee and human, there are many more. Right? So that gives an idea that these are the changes essential for our brain development. And in fact, without genetic change, there is no evolution. When I talk about evolution, genetics play a major role. I want to highlight this paper published by Dibjansky. The title of this article is The Raw Material of Evolution. So what he was mentioning, the raw material is nothing but the mutations. The mutations or the genetic changes are the raw material for evolution. And in this very first sentence he has made, I'll read that. for nearly 80 years, evolution has been one of the basic problem of biology. What he was mentioning, of course, you can see when this paper was published, 1938. The 8 years what he mentioned was the book which Charles Darwin wrote in the year 1858. The title of the book is The Origin of Species. Okay. He was talking about that. I hope we all believe on evolution. All of you believe? There's no one who don't believe on evolution. No, nobody believe? Okay, only two. Okay, few more. Even if you don't believe, I'll give you some example. The mutation in our genome has brought the phenotype the way we look like today. Okay? Look at these three brothers who have born to a normal parent. Dr. Anuradha mentioned about the recessive mutations and so on. I'll give you more example with our own findings. So these three brothers born to a normal parent has the typically like the phenotype of chimpanzee. Okay, that itself shows that the mutation which we gained during the course of evolution for the last several millions of years, it has gone back. This is a back mutation, therefore they appears like a chimpanzee. Okay, this is one. Second example, during the course of evolution, we all lost the air on our body, but look at the child, because again back mutation, the air has come on the child. Okay, this is a second example. Third one, we all walk with two legs. Chimpanzee walks with four. Right? So this is a Turkish family. Somebody is interested, they can see this YouTube video. There are 5 children. Elder son only can walk with the two legs, but again not with the steady walk. He walks like a drunkard person. But the 3 or 4 sisters cannot walk with 2 legs, they walk with 4. This is one of the ability which we gained. There was a back mutation which again taken them millions of years back. So all of these are good example for the evolution and the mutation in a genome play a major role. These are some of the other genes which are involved in human evolution. When we talk about brain, myosin heavy chain gene, again remember the gene is highly conserved, this particular gene or many more genes. But the mutation the gene is the one which brings the phenotype. This particular gene where the mutation created help in creating more space in our skull, therefore the brain size has increased. There are several other genes which are responsible for increasing the brain size. This particular gene, buprenorphine, that is involved in learning, experience of pain, perception, behavior and memory. So these are some of the very unique features of human. Right? So again mutation this gene has gave us so much of advantage. Even for our speech, FOXP2, some of the genes are involved in making our jaw muscle very loose, therefore the speech have evolved. So coming back to the slide that I told about the human evolution, and the human and chimpanzee have diverged from a common ancestor called hominids. Can you guess how many millions of years back? I told you, it's about 6 to 7 millions of years back. Right? So during this 6 to 7 million years, there are several species have evolved and all of them have vanished except human. And this process, there are several features we gained. One I already mentioned, walking with the 2 legs. Other one is making tools for hunting animals for food and so on. Third one is the brain size, which also we have discussed. And if you look at the brain size in the early species, the chimpanzees, brain size was as similar as chimpanzee. It has increased to 35% of the modern human, the way amount which we have now. In Homo habilis 50%, Homo ergaster 60%, it has gradually increased. And you all know very recently, a lot of talk about Neanderthals. The brain size was predicted that it was slightly larger than human, okay? But nobody knows whether they are intelligent than human or not. But there was evidence, which I will tell you later, that human Neanderthals have lived together. Okay. But why this species vanished? There are many interesting hypotheses. One of the hypotheses given by Paul Mills from Cambridge, he said that inherent violent behavior of modern human probably responsible for vanishing of the species. May be true, because we all know how violent humans are. Anyway, so I want to give you a little bit about Neanderthals, and this was found mostly in Europe. These are the places the Neanderthal bones were found. There was extensive work was done very recently by a group led by Svante Pääbo in Germany, and the first time very successfully you could be able to isolate DNA and sequence. There are about 40,000-year-old bones of Neanderthals. And because he had done very extensive work on this, demonstrating that one could be able to isolate DNA and make use of the DNA for our evolutionary studies, and about 3 years back or 2 years back, he has been awarded with Nobel Prize in the field of medicine or physiology. So you can imagine that how much of work he has put in this particular field of paleo genetics or paleo anthropology and so on. So now we have seen human and Neanderthals are very close, maybe 500,000 years apart. But you can ask question whether we all carry some amount of Neanderthal genome, because when you believe on evolution and Neanderthal is very close to human, we all should carry some amount of DNA, right? So in one of the study where we're all part of it, we sequenced 300 human genome across the world, and the study was led by one of our collaborator, David Reach, where Fabo and myself and Dr. Lalji Singh were part of it. And we have shown that people who are living out of Africa, that means zero, that means African doesn't have Neanderthal genome, but people living outside Africa carry up to 3% of Neanderthal genome, suggesting that yes, Neanderthal is very close to human, and it gives some kind of evolutionary bridge between human and other Homo species. Okay, let us now come to origin of modern human. Again, you all must have studied the modern human arose in Africa about 160,000 years back, and from there migrated to many parts of the world. And the question is, who is the first out of Africa human? Could anybody guess? And they are in the Indian subcontinent, right? If you are unable to guess, I can show you that this picture. The Andaman tribes are the very first modern human who have migrated out of Africa. I think most of you are surprised to see there is a population like Africans are in India. There are 4 tribes: Onge, Great Andamanis, Jarvas and Sentinelese, in 4 different islands of the group of Andaman and Nicobar Islands. While Onge, Jarva and Great Andamanis have some contact with others. Sentinels are still in isolation. Nobody can go near them. They will shoot with arrow. It happened multiple times. Okay. So, we had an opportunity to go and collect the samples after obtaining permission from government, Indian government and local government. And of course, there are several steps to go there, and we had fortunately went and collected the samples. And we use different markers. Sometimes use selected markers, sometimes use whole genome, and it ranges. In a given cell, let us take somatic cell, you all must have studied. There is a 46 chromosome in human, 23 we inherit from mother, 23 from the father in the form of egg and sperm. In addition to that, we also have mitochondrial genome. In a given cell, there are 2 copies of nucleus genome. There are multiple copies of mitochondria, and each mitochondria have multiple DNA. That means in a single cell, you will have hundreds of mitochondrial DNA, and all of these are inherited only from the mother. So, you all know that mitochondria is known as a powerhouse of the cell, right? And you can see where we all get the power, only from the mother. Okay. And therefore, using mitochondrial DNA, we can trace the maternal lineage. Of course, this is also associated with a large number of diseases known as mitochondrial disorders. Earlier, it is known that mitochondrial disorders are transmitted only from the mother, but now there are large number of nuclear genomes, nuclear genes are also involved in transmitting mitochondrial disorders. So, we use different kind of markers. We use Y chromosome marker for tracing the paternal lineage. Only the males have Y chromosome, and Y chromosome and mitochondrial DNA are transmitted uniparently without much change. Therefore, these are the 2 markers used for evolutionary studies. So, we used the mitochondrial DNA on these tribes, Andaman tribal groups. Initially, we looked at only a small proportion of mitochondrial DNA, and then we sequenced the complete mitochondrial DNA. In fact, these 2 studies have helped us in coming with the conclusion based on the mutations which are present in these 2 the Andaman tribes, and these are the mutation position on the mitochondrial DNA, and these set of mutations are not present anywhere in the world. It is present only in Andaman tribes, and based on the mutation we can also calculate the age of the population. That helped us in proposing that the Andamanis are the first modern human who have migrated out of Africa, taking southern coastal route and reaching Andaman and Nicobar Islands about 65,000 years back. And this is a very first genetic study suggesting that the Andamanis are the first modern human who have migrated out of Africa for the very first time. Subsequently, after human genome was sequenced and about 1 million SNP single nucleotide polymorphic markers have been identified, and we used that 1 million genetic markers, and we selected about 25 populations representing all the 4 major linguistic groups. All of you are aware we have 4 major linguistic groups: Dravidians, Indo-European, Austroasiatic and Tibeto-Burman. Again, under each linguistic group there are several languages. For example, if you take Dravidians, we have Tamil, Malayalam, Kannada and Telugu. Similarly, all of them have multiple language. We also carefully selected the social group, the so-called caste system and population across India. And this study, using 1 million genetic markers, helped us in proposing that in the prehistoric India, there were two founding populations. One we call them as ancestral South Indians, other one is ancestral North Indians. Okay. While the ancestral South Indians are part of the early human migration, when the Andamanis were migrating from Africa through coastal route, and that journey actually helped some of them staying back in southern part of India, whom we call as ancestral South Indians. Some stayed in Andaman and Nicobar Islands, some further migrated to Australia. The Australian Aboriginals also have genetic affinities with the Andamanis. On the other hand, there was a second wave of migration from Africa towards north, and somewhere in the middle, maybe in the Middle East, there was a division. One group has gone to Europe, other group has come to northern part of India, whom we call as ancestral North Indian. There are a large number of evidence to show that Europeans and ancestral North Indian have some kind of genetic affinities. That includes Y chromosome haplogroup, the skin color mutation, because everybody, if started the journey from Africa, why some of them are lighter? That is because of the vitamin D deficiency, and those who have migrated to Europe, they might not have seen the sunlight for many weeks or months, and they must have suffered because of low sunlight. Therefore, the mutation introduced in a gene called SLC24A5, so that turned the skin color into lighter. The same mutation is present in Europe, Middle East, and also northern part of India, and there are several other mutations. And these two groups have admixed and gave rise to many populations, which we will see in the coming slides. On the other side, if you look at, these are all Indian population. This is called as principal component analysis. There are many individuals here, many Indians along with Europeans, Africans, and Chinese. There are hundreds of individuals in this, where all of them are in a single cluster, suggesting that genetically they are more homogeneous, not much difference. Whereas look at every single population in India is very unique, but although every single population is unique, but still we share some proportion of genetics with other populations. That's the reason there's a gradient of clusters. Right? We're all different, but still we all share some kind of genetics. So best example that we all admix is the skin color alone. If you go to extreme north, like Kashmir, people are with very light skin color. If you go to extreme south, I think some of the tribes in Kerala, including Kurumba, they are with still dark skin color. These two are too extreme, but in between there are so many shades that itself shows that all Indians are admixed. Okay? That is what is given here. So that means India is inhabited by large number of small populations. To understand this particular phenomena of admixture between ancestral South Indian and ancestral North Indians, we took another study where we included large number of population from north, large number of population from south. Based on the recombinations, we predicted that in the prehistoric India, as I mentioned, two founding populations, and each population gave rise to many population groups. You can imagine that the first out of Africa human, while migrating, some groups settle in south in a jungle. And maybe after few years or few hundred years, after completing whatever the food material or animal available there, and some of them, those who are active, must have migrated to some other place, from there to other place. That's the reason there are so many populations have evolved from these two founding populations. This had happened until last 4,000 years. For the last 2,000 years, these two founding populations have admixed. you can see the color just as an indication that the genome is admixed between these two founding populations. After admixture, then every single population started maintaining the endogamy. Endogamy means marrying within the group. So now what we follow that every caste, right, every tribe marry within the group, but the caste system came much later, maybe around 1000 or 1500 years. But the population specific marriage was even older than that, maybe around 2000 years. Okay. Now let us see, because this is the culture I was saying, that practicing endogamy, and let us now look at what is the impact of that endogamy on our health and disease. To understand that, again we took another study where we used 2800 individuals from 275 population. So this time we do not want to restrict with only India, we wanted to take all the South Asian populations, including Sri Lanka, Pakistan, Bangladesh, Nepal and so on. The reason why we wanted to take other South Asian population is that whatever we do in India, it is also relevant to other South Asian populations, particularly when you are looking at genetic mutation which causes some disease, which is highly prevalent in other South Asian countries. I will give you just one example. One of our earlier studies where we found a 25 base pair deletion in a gene called myosin binding protein C3. Okay. This mutation is not even an exon, it is in the intron. Right. Intron is known as its non-coding. Right. But the intronic mutation skips away wild type exon, therefore there is a truncation of the protein. And this mutation, when it exists in the heterozygous condition, that means two copies, we have 46 chromosome exists in 23 pair. One of the copy has the mutation, other copy is normal. The individual survive up to 40 or 45 years without any problem. Then when they are very active, like athletes or cycling, then they collapse. But if the mutation exists in the homozygous condition, the children die at very, very early age, less than 10 years. By looking at the postmortem tissue of the children who have died at very early age, we found mutation exists in the homozygous condition. It causes three different kind of cardiomyopathies. One is hypertrophy, thickening of muscle including an interventricular septum, dilation, the ventricle enlarges beyond the normal size, and restrictive, the heart muscle become very stiff. Therefore, it suddenly stops pumping the blood, therefore, there is a sudden cardiac arrest. When we try to look for that mutation in large number of Indian population, we found it is existing at 4.5% all over India. Of course, different, slightly different frequency in different state, but on average 4.5%. When we try to look for its frequency all over the world, but to our surprise we found it is present only in South Asian countries, but not in other countries. And this mutation also we predicted it arose somewhere in India about 33,000 years back. And because of the previous slides, if you recollect, there is a population admixture, there is a movement of people around the world. Therefore, it got well spread across India and also other South Asian countries. Okay, now I am coming back. This is to tell that why we included the South Asian population also. When we study these 275 populations, we do not have any clue at all whether they have any disease or not, because they are all collected for population study. When we did various statistical analysis, this is one of the analysis called as IBD score, means identity by descent. And if the length, every line represents one population, there are 275 lines. If the length of the line is very high, that means the IBD score is very high, and the impact is, in a simple term, if you are analyzing 100 unrelated individuals from a particular community, and if you calculate the IBD, 90 out of 100 having the same haplotype. That means there is no change in the DNA. Maybe you can take 10 kb or 100 kb. That suggests that all the 90 individual must have come from a common ancestor. Okay, they will have identical sequence. But out of 100, only 10 of them having the same haplotype, other 90s are different, they will have low IBD score, they may fall somewhere here, right. But this slide also included two populations who have been well studied in terms of genetic diseases. One is Ashkenazi Jewish, other one is Finnish population. You can see they are very tiny, they have tiny IBD score. For that itself, they have so many diseases, most of them are autosomal recessive. Even this Finnish, this is a Finnish population, this is Ashkenazi Jewish. Now you can imagine with the very tiny IBD itself, they have so many diseases, and what would be the consequences of large number of Indian population who have several fold high IBD score, right. One can predict that yes, there could be large number of population specific recessive disease must be existing in Indian subcontinent, right. As Dr. Anuradha also mentioned about the heterozygous parent and homozygous children and 25% of chance and so on. So, this pedigree exactly shows that. So, what is the problem with the recessive disease? If it is a dominant, heterozygous will have the disease, right, no issue at all. Whosoever carrying the mutation, immediately you can see they have the disease. But the recessive mutation problem is if it exists in the heterozygous condition, as you see here, there is a mutation here known as the pairs. She is normal, that pedigree I think all of you are aware as a genetic students. She married somebody without that mutation, both the chromosomes are with wild type, and this couple have two daughters, and again this mutation going in heterozygous condition, both the daughters, and they also married somebody from outside without the mutation. Those therefore, there is no issue at all until here, right. This mutation goes generation after generation, right. It can go even further. But now these two must have lost their relationship because after so many generations. Now, if these two marries, as Dr. Anuradha described earlier, there is a 25% of chance that the mother contribute this chromosome, father also contribute this chromosome. Therefore, become homozygous, therefore, there is a disease. So, that condition is very, very high among large number of Indian population, where we do not have any epidemiological data, right. So, to understand this, we thought, okay, let us check few population who have high IBD score from southern part of India. So, we went and collected the samples, and in this study we found large number of population specific mutations. We used 4 populations, right, and in this we had population specific mutation in 3 population. One in Andhra Pradesh, high frequency of ankylosing spondylitis associated with HLA B star allele. Another population, again very rare unique mutation associated with cardiac disease. We are characterizing that, but now I wanted to highlight very important mutations where we did little extra work and reached to the level to screen the population. So, one of the population where we found there is a disease called junctional Herlitz epidermolysis bullosa. That is a skin disease and also affect internal organs, including it lead to congenital heart disease. Okay. So, the children unfortunately they do not survive beyond 2 months. Within 2 months after the birth they die, right, and no information so far. Therefore, we sequence the exome of the child and their parent. We found there is a 11 base pair deletion in a gene called LAMB3. The parents are heterozygous. Okay. The children with the homozygous mutation with the disease, they die within 2 months. This is a novel mutation. So, recently we sequenced 10,000 human genome. In fact, that was announced by Honorable Prime Minister last year. We looked for that particular mutation in 10,000 individuals from 83 communities. So, none of them carrying this mutation. Okay. Then we went on to check in the global database consisting of 8,62,000 individuals, and if you convert into chromosome 17 lakh chromosomes, we found only 2 individuals from South Asia origin have heterozygous. Otherwise, none of these 17 lakh chromosome have this mutation. That means it is so rare, present only in one particular population. Right. If the mutation is so rare, we are not very sure whether this mutation is real cause for the disease or just kind of association. Right. Then we looked at the protein structure and the amino acid sequence. The full length protein has 1172 amino acid. Because of the mutation 11 base pair deletion, it truncates, it introduces a stop codon at the position 799. That means it removes 373 amino acid. Therefore, the protein is very short. Therefore, it is unable to do its function and leading to the disease. This is a prediction, right? How to prove it? We need to create animal model. You all know about CRISPR-Cas technology, right? You can precisely delete a base or whatever the size you need by designing guide RNA and so on. I will not go into detail. So, we precisely removed this 11 base pair and made an animal model with this strain, and these are the pups. But what happened in the human, the children are not surviving beyond 2 months. Here the pups are dying within 2 days after the delivery. So, how to proceed it further? But within the 2 days, we could see there is a sign of scaling on the skin. Probably it will lead to further skin rashes and so on. We also looked at the internal organ by doing micro CT. CT all of you know, but this is a micro CT for the animals. Looked at the heart. This is a wild type, no deletion at all. This is a heterozygous, one copy is normal, other one is with the deletion, and these two are almost the same. There is no difference between the wild type and the heterozygous. Whereas the homozygous, where both the copies having 11 base pair deletion, you can see hypertrophy, the interventricular septum is so thick. Also there is a septal defect, there is a hole between the septum. Therefore, that may be the real cause for the children in human dying within 2 months and the pups also dying within 2 days. Okay. Because it is dying within 2 days, we do not have any clue to see the real phenotype. Therefore, we went on to introduce different set of mutation. One mutation which we deleted was 45 base pair within this region, that includes 11 base pair also, but this is a non-frame shift deletion where the original 11 base pair introduce stop codon which removes 370 amino acid, but this, although the size of the deletion is 45 base pair, but it removes only 15 amino acid, therefore the impact is not so severe and the animal was fine until the 4th month. Then slowly it started showing the symptoms, and this is after 4.5 months. You can clearly see the same phenotype what we have seen in the children, also in the mouse, suggesting that yes, this is the real cause for the deletion, cause for the disease. And the internal organs also affected, and we looked at carefully the internal organs. The heart, you can see there is a enlargement, spleen also enlarged, the stomach become very small, and later by looking at the micro CT there is a lot more abnormality I have seen. I am not going into detail of that. Then we looked at the skin. In the wild type you can see there is no the distance between dermis and epidermis, it is intact, but here you can see there is a gap between dermis and epidermis. That is where probably the blister starts and gets spread across that. Then we also wanted to understand in the human, because we do not have a child or children surviving with the homozygous mutation to study. We thought of introducing homozygous by taking the amniotic fluid of the mother, which has a heterozygous mutation. In the normal chromosome, we try to delete 11 base pair. Okay, now we made homozygous line from the mother's amniotic fluid, and we tested with all the pluripotency markers and are all fine. Then we set up the experiment to see, we transform that into skin fibroblast, and this is a heterozygous with this simple hematoxylin eosin staining, and these are keratinocyte marker. You can see absolutely skin structure is fine. On the other hand, in the homozygous mutation, you can see that the structure is not fine. Okay, this is one experiment. We also try to look at the heart, because heart also we found abnormality. We again transformed into cardiomyocytes and found in the heterozygous condition the iPSC cells with Lambda expression. You can clearly see there is a red color showing Lambda expression, and here you do not see much of expression, very mild, right. Further we went on to see how the group of cells, where you can also see this when you are growing the cells, you can actually see the heartbeat kind of thing. This is the heterozygous, you can see there is a beating of the cells, whereas in the homozygous you do not see much of it, very very mild and has been captured. You can clearly see the rhythm of both. All this suggests that the 11 base pair deletion is responsible for all the phenotypes, right. So this is basically for our understanding. Of course, lot of other work is going on, transcriptome, proteomics, we are looking at the sperm abnormality and many more things. But these are all for our understanding of basic biology of the mutation. But with this, can we do something for the families? Okay, we keep on asking, is there any pregnant woman who have lost the children previously because of the mutation? And we found there was a mother with a pregnant who lost a couple of children. This basically near Madurai, this family, and we brought the mother from Madurai to Chennai, collected the amniotic fluid, and we did simple PCR based test to find out whether the amniotic fluid is with the heterozygous mutation or wild type or homozygous. Fortunately, when we tested, this is a homozygous single band heterozygous, and amniotic fluid consisting of 2 bands. Further we sequence and confirm that is heterozygous. Therefore, we advise the mother to continue with the pregnancy, and it is our joy to see about 1.5 years back the child born with normal phenotype, and we keep on watching, finding out how the child is doing. She is fine, right. So, this gives some kind of satisfaction that the work which we started for understanding population history has gone into different direction. Now, we are in a position to identify a population specific mutation responsible for population specific disease by predicting based on the genetic variation, right. So, this is up to a family, but now because this mutation is population specific, we want to do beyond that. So, we wanted to screen the entire population. There are about 5,000 individuals, but we can't do without government support because entire population. Therefore, we approached Directorate of Public Health and Preventive Medicine, Government of India, to give us the approval to screen entire population. And after about 1 year of paperwork, there are several committees we will have to present. Last year, we got the approval from the Directorate of Public Health and Preventive Medicine. So, therefore, with that, we went on to do the screening with the proper strategy. We have 2 different strategies. One, with the pregnant mother, as I explained, we do the screening, and based on this counseling, decision has been made. But in the normal individual, we want to collect sample from every single individual, do the same way, and we want to recruit somebody from the same community, use them to create kind of database to store all the information. Okay. For example, now we are recruiting school children, they are 5 years or 10 years, and if we found heterozygous mutation who is absolutely normal, he or she grows maybe after 10-15 years, and he or she want to marry someone, and they can take the database information and see whether that person, because this population is so highly endogamous. They are not even going to the next district. There are 5 different districts, same population, but this mutation was found only in one particular district, right. So, they are going to continue that, at least using this information, they can give appropriate counseling. Therefore, this mutation can be avoided by spreading into the next generation. So, therefore, we can in fact eliminate this mutation from the population itself. So, we made a pamphlet in both English as well as in Tamil, local language. We went there, had a camp last July 19th and 20th, was explaining them in the local language, Tamil, and a lot of people have come, and we collected the samples, and we found actually 8 individuals who doesn't have any clue, doesn't have this mutation in the future generation. 8 of them, accounting for approximately 4.5%, are with heterozygous mutation, right. I am sure that is going to be across the population. Now, you can see the impact of this study, and is going to be very, very useful for the future generation. And this is, if you set this as an example, made a model, and we are going to utilize this for large number of such populations which are carrying a population specific recessive disease in the future, is going to be very, very helpful for the society. So, with that I stop by thanking all those who have contributed to the study, my colleagues and large number of collaborators, and funding is very important, has come from various sources: Genome India, Pediatric Rare Disease, CSIR, UKRI, DBT, DST, and Medical Research Council at London. So thank you very much.
Now we'll move on to the questionnaire, most awaited thing. Please, I would like to invite Dr. Sumanlatha Madam, Anuradha Madam and Thangaraj Sir onto the dais. Questionnaire session. So please shoot your questions in orderly manner and please specifically tell your questions to those who want to question it. Please tell your questions and address the speaker. Students.
So, I have a question for Anuradha Madam. So this is about when you present a report, a genetic report, whole exome sequencing, mostly the genes which are in the database like ClinVar, they get reported. But sometimes there are also variants like variants VUS, uncertain significance. They don't get reported, but you will have to ask them for the data, right? Whether you can have access to the raw data, and then you have to do the analysis and find out there is this problem. And a lot of time goes from the original report and your personal analysis. So how do you, because you are in this field, how do you plan to address this problem? Because nowadays we have AI like ChatGPT, which are quite powerful. You can give them the VUS, and then it tells you, yeah, maybe this is something you will have to go forward and investigate. Yeah.
Good name?
Yeah, I'm Siddharth, and I am a faculty in Aurora College. Yeah.
Okay, okay. So, very nice question. Variant of uncertain significance is a challenge to genetic laboratories when we are reporting. So, everybody has their own guidelines. There are international guidelines, and there are also guidelines that each lab would develop on how and when a variant of uncertain significance can be reported. Because all of us will have hundreds and thousands of variants, and you cannot be reporting everything. So the number one guideline would be if there is a phenotypic correlation. For example, if you are seeing a child with epidermolysis bullosa or any other genetic condition or a phenotype that we would call syndromic or non-syndromic presentation, and if the variant that we are looking at has a relevance to the phenotype, although not reported in past in literature, although the pipelines or in silico predictors are saying that it might not be disease causing, we have to report that variant of uncertain significance because of clinical correlation. That is one. If there is no clinical correlation at all, then variant of uncertain significance are not reported. In carrier screening also, we follow something similar, but then you don't have a child already born with an abnormality. So that is even more complex, whether to report variant of uncertain significance present in both partners, which might result in an abnormality. We don't know in future, right? So in carrier screening, we do report variants of even variant of uncertain significance has a leaning. Some variants which are uncertain lean more towards pathogenicity, some lean more towards benign. So looking at the what is it that it is leaning towards, we decide what to report and what not to report. And we do save the data for a very long time, so that because a variant of uncertain significance today can become benign after 2 years, or it can become pathogenic as per the literature and databases. So we look at that, we make sure that the data is stored, and it can be used for reanalysis. All exome sequencing reports, we advise that a reanalysis can be performed after a couple of years.
To Madam Dr. Anuradha, GeneTech actually. So recently I have come across an article, a news, exciting, that a sperm from oligospermia is taken using artificial intelligence. They selectively fertilize the egg. So what could be its future in India? Is it possible to move in that direction? If it is possible, how?
Sir, AI utility in infertility has increased tremendously. So what they used to do in the laboratory under the microscope, a lot of it AI is doing. They're able to select the best of best sperms. And now the sperms don't even have to swim. You can select the sperm with a bent flagella or even the acrosome which is required to penetrate the egg. Even in those absence, you can simply take the DNA, the sperm, because the DNA is intact, and you can do a ICSI procedure, in vitro, the cytoplasmic transfer, and then you can fertilize. AI has a very, very big role in infertility, because it is not only it's able to pick up even embryos and eggs of good quality, because naked eye is naked eye. You see the morphology of the embryo and you say, okay, this is A1 embryo and I can keep it. But then the AI is able to predict, the surface, the inner core, the way it is growing, and then it is able to suggest which is the best embryo to transfer. So AI in terms of imaging combined with genetics, we are able to do, the success rates of infertility are going to be really very high.
Dr. Vanaja from IICT, ma'am, Biochemistry Department. I completed from that. My question is, nowadays most of the women who are affecting before marriage, their posture is changing with the HCG hormones, injections. So how can we stop this, ma'am?
Can you please repeat?
HCG hormone.
Hormone?
Growth hormones before, when they conceive, they are giving the injections. So before marriage, their posture is changing. After conceiving, the posture is changing. Yes.
Okay. So things are, I'm not an infertility specialist actually,
Yeah,
but I do work with a lot of them, and I know that even in that area, things are improving quite a bit. And there is this thing that you take lot of injections and then it puts your BP increases, your weight increases and things like that. But see, that happens because most of the time it's a trial and error thing. If something works this cycle, it doesn't work, then they go for another cycle with some other change in the protocol. But now, as I was saying, with genetics and with a lot of other improvements within fertility medicine, the number of cycles are drastically decreasing, and they are able to get better eggs with less number of stimulation. So the side effects are going to be minimized day after day. Although, yeah, it's not going to be like natural conception, but I think there are quite number of improvements that are happening there.
But the posture is changing, ma'am, before marriage and after marriage.
Posture, you mean?
Yeah. They are becoming obese.
That does not happen for each and every patient. I mean, because there are so many other factors, like whether the person has a PCOD or not, and whether they have any diabetic condition, pre-diabetic condition or not. So based on that, yes, I do understand that for some people, even stimulation causes what is called ovarian hyperstimulation syndrome, which is a very dangerous health situation. But I would say in last 10 years, they have reduced ovarian hyperstimulation syndrome from around 5% to less than 0.05% today. So there must be few cases where these things happen, but otherwise it is preferably, fairly safe.
Thank you, ma'am.
Thank you.
I am Dr. Rajendra Rao. Yeah, the time for the era of the AI. So any young minds are here. So can you elaborate more? How can I use the AI in our genomics to take it forward?
Repeat your question. How to use it in
the era of the genomics and sequencing era.
So AI, although now we are talking a lot about it, at small level, machine learning has been used for calling variants. Right? I think some of the companies, including Illumina, so their own software like Trajan, it uses manually. If you use a computer for extracting the variants, It takes several hours or sometime days. But for exome, you can just get all variants in 15 minutes. For the whole genome, maximum half an hour. Right? So that's a way of AI and ML being used. But now that will be even more faster. Using AI, you can predict drugs based on the genotype. We don't have to do a lot more experiment. Using AI, you can just quickly predict what we call as tailor-made drug for a specific mutation. Right? So all of these are now possible, and therefore I think government is now spending a lot more time and money in having AI in the healthcare sector.
Also in the something that I thought would be of very interest to especially students is AI development is happening in the area of pediatric dysmorphology. So if you have a child with who is not showing the features as normal as we would imagine, you can take a picture and the picture will collect all the measurements of the face and it will, or of the body, and then it will tell us that it probably could be this syndrome, and this syndrome. And this happens in a few minutes. And so these type of tools are helping geneticists, they are helping pediatricians, they are helping all medical communities to come to diagnosis as quickly as possible and also do what is called a differential diagnosis. So when you look at someone, something, any problem, you feel like this is the problem. Your mind kind of fixes on it. But then having these tools help you to do differential diagnosis. It will say it could also be this, and it will give us an opportunity to do better diagnosis.
Just add to this, some of the pathological investigation, including breast cancer identification, sometimes by regular or conventional histopathology, pathologists can miss some place, whereas AI, with very precisely, it can predict some of the place where there's a cancer. So now it's been used very significantly. Of course, with the caution, almost everything is based on the how much of input we are giving, right? If we don't give enough input, sometimes it could be wrong also. And one incident I can say that I think that recently I heard a talk where they were chatting with AI and it has given almost right answer. Then finally the question is, how did you know? The AI say, I have a degree in public health. Right? So that means somebody has fed that information. Those who have done the public health has provided the information.
So in time or shortest time, precision in resolution also matters a lot with
that
AI.
Is what it is.
Yes.
Students should ask.
Students back.
Good morning, sir. Good morning, ma'am. I actually want to ask, is there any internships provided from the GeneTech for BSc graduates to enhance a hands-on experience or everything like?
Okay, good question. Yeah, yes, we do provide internships, but they are very limited, ma. So our lab is a small laboratory. We can accommodate at any time not more than 2 interns. So always there are 2 interns who are working, we will try and accommodate more. So if you want, you can just go to our website, find an email, there's an info@genetech.co.in, send your resume and request for internship, and there will be an interview, and if you are selected, we will be more than happy to have you around.
Thank you so much, ma'am.
Thank you.
One more good.
I
have a question for Dr. Kumaraswamy Thangaraj, sir. Firstly, good afternoon, sir. So, I want to know why Sentinelese tribal people resist contact with the modern world.
Sentinelese.
Yes, sir.
So, because they have their idea that whosoever is not belongs to them is dangerous to their community. Right? So, that has been prevailing for all the 4 communities. The same thing is true for Jarvas. What happened for Jarvas recently, the government laid a road, therefore some contact was made, but still they were not accepting the people. One Jarva child met with accident, he was on the road. One doctor from GP Pant hospital took him to the hospital, treated and went back and left the same place. He acted as an ambassador telling that no, as we think they are not enemies to us, they are friendly, they treated all those things, then they become slowly coming to contact. Right? Until otherwise they believe on others, it's very difficult. Yes.
Thank you so much, sir.
Okay.
Sir, my question is for Anuradha ma'am. Ma'am, before that I would like to say your journey was quite inspirational, and as you mentioned earlier, if you are treating someone, you want to find a problem, but other than you also find another problem. So, in your opinion, will you tell that patient or will you keep it as a secret?
No. So, first we ask the patient whether they want to know such incidental findings or not during the counseling session. If it's not a counseling session, we make a call and we ask them that we are releasing the report, would you like to know? And they will ask what will happen if I have it and things like that. So it requires a little bit of communication. And once they say yes, then we only report pathogenic, that means disease causing variants which increase risk. So we don't report anything with uncertainty. Okay, we report only those which are contributing to the disease. So as per guidelines, there are around 80 genes for which can be reported and where preventive management is available. So we report those genes and then we recommend preventive management.
So that's also just add some point to this. Sometime we need to be proactive also in very rare cases. For example, somebody who is involved in public public transport, for example, driver of the bus, and incidentally find that there is a mutation which causes seizures, right? So we cannot leave it like that, right? Because 50-60 people's life is involved in that. So in that case, we have to proactively say, therefore the treatment can be done or something. So we have to look at case to case differently.
Thank you, sir.
Myself Arzafnan from Aurora College. I would like to address to Dr. Sumanlatha. Can misuse of antibiotics increase mutation in TB bacterium?
Yeah, definitely it will. Mutations.
Like in what ways?
Like different drugs, what they are using, if they are not using properly, then there is a possibility of the mutations.
Thank you.
Relapse.
Priyan from Aurora College. My question is limited sources having a big ambition for opening a company like GeneTech. What will be your advice to create an impactful company like GeneTech?
Crying, but yes, please go ahead and do something of that nature. You please aim high. It is always very you always end up highly satisfied if something of your own grows, like your child. My children say that the GeneTech is my first child, followed by 2 daughters. So I have 3 children. So the happiness of establishing something, growing something, and seeing success is something that is not describable. So if you have the grit, if you have the potential, if you have patience, you should go for it and do something of your own. You will have problems, but you will overcome it. Yeah, it will give you immense satisfaction. Please go ahead.
Good afternoon to Dr. Dr. Anuradha Ma'am. This is a question following on that question. Is there a right time or the right amount of knowledge a life science student should have to become an entrepreneur?
So it depends. I mean, what do you want to do? For example, I always felt that I should have had an MBA, because I did not even know proper difference between credit and debit when I started my company. And I always thought I was very, very poor at finances, and if that kind of if I had that knowledge, it would help. So you have to think like what time you want to become an entrepreneur. Do you want to, in which area, and how much education do you require to be able to do that? Of course, you can hire people with that kind of education, but what is your idea? What is the need in the market? Have you done the market analysis to say that you will be able to grow in that area? How do you manage your finances? So you have to figure out all these things and then become an entrepreneur, just not jump into it. So you have to have a need, right? At that time when I was establishing GeneTech, there was no private company which was offering genetic diagnostic services to pay doctors. So I knew that there is a gap there, and I have established that. So first there has to be a need, and then the market should be mature enough. When I have established GeneTech, my market was not mature enough. So I had to create what is called awareness in the market. So are you ready to create that kind of awareness, or is there awareness but then there is a need or a gap? In such a scenario, awareness is not your problem. Then you can establish. So you have to see what area you want to establish and how much of, and of course you have to look at the investment. How much of investment will you require? Do you have funding? Do you want to put in your own money, or do you want to get investors into it? And how do you project? Because if it is entrepreneurship, you are obviously doing it for profit, right? So you have to have a 5-year plan. So there are many things involved. There is no correct time. I mean, I can start a company today again. So you can start it today if you want to. So there is no time. It just has to be the right time, and you grab that opportunity and do it with full focus.
Thanks, ma'am.
You're welcome. I want to see at least 5 or 10 entrepreneurs from this room. That will be great for the country.
Good afternoon, ma'am. Good afternoon, sir. My doubt is, in case when closely related individuals wish to marry, how can genetic screening help identify and prevent mutations and inherited diseases? How can counseling help them?
So that's what we do. We call it carrier screening. Counseling helps a lot. These days people are coming before marriage. So we call it pre-marital counseling. Someone wants to marry their cousin, then both of them come, we take, or someone want to start a family, they already married a cousin, then they come for counseling. So on a theoretical level, there is 25% risk for a genetic disorder only if both of them are carriers of a specific mutation that causes a disease. So, but there are 25,000 genes. They can have disease causing mutation on any of these genes. So we now have the technology and ability to be able to sequence all these 25,000 genes, identify if they are carriers or they share any common mutation in any specific gene. Then we will know, and we can tell them that, look, you have 25% risk for thalassemia or muscular dystrophy or some epidermolysis bullosa. And then you have options. what options? You can either test in your embryo or you can test in your early pregnancy.
Thank you, ma'am.
You're welcome.
Good afternoon to the ones on dais and everyone present here. I'm Yogita, faculty at the Indra Pradarshini College, Nampalli. So this question is to Dr. Thangaraj sir for the Genome India project. As being an eminent person and a leader in the Genome India initiative, what do you hope to achieve with this comprehensive genetic map of Indian population? Should I repeat myself?
No, this is fine.
Yeah, so followed by that, I just want you to also add on your current work, which is about what is your current focus on your rare pediatric genetic disease?
Yeah,
Thank you.
very good. Yeah. Look at global database. Actually, we don't have a large number of Indian genome sequence. You can sit. I think about 2 decades back, there was opportunity, but we could not be part of it. But now, cost has come down, and the funding agency also enough fund to fund it. So this is the first time we sequence 10,000 Indian genome with the idea to catalog variations in this 10,000 individual belongs to 83 populations, and we don't have having the reference genome for Indian population. So one of the complexity which I mentioned is we have 4,635 populations, right? But every population is very unique. So if you wanted to use European genome as a reference sequence, we will miss a lot of information. Therefore, we need to have our own genome as a reference sequence. Therefore, even somebody is sequencing very low depth, they can use that panel and use our own sequence to find out the missing sequence, because if you look at the haplotype wise, there could be a mutation which is Indian specific, may not be existing in European, but when you are using European data as a reference, you may pick up a wrong variant. So therefore, it's important for us to have our own genome as a reference. So that's the second, cataloging reference sequence. And this will also helpful for us to find out how some of the variants which are present in a heterozygous condition, which are recessive in nature. Okay, that is second one. We can also find out the variants which are present in drug metabolizing enzyme to identify whether certain drugs are useful for Indians or not at all. Right? I'll give you a couple of examples in this line. When we looked at the 10,000 genome to look for a specific mutation, which so far we know that is present only in Vaishya community, that is in the BCH-E1 gene. Because of that mutation, when butyrylcholinesterase is given during the anesthesia, this is a muscle-reluctant drug, they cannot metabolize that. Therefore, those who have the mutation will remain with the anesthetic effect for very long time. Sometimes it leads to paralysis, even death. Right? So far we know that is present only in Vaishyas. But when we try to look for this variance, it is present in 29 other population in a heterozygous condition. This is also recessive. Right? So the amount of information is giving is that we don't have to, it's not that only we have to look for the Vaishya community, whosoever is having the mutation, screen before the drug is given. We also look for the other 29 population. Now it's heterozygous, but it may eventually become homozygous after a few generations. Therefore, they also face the problem. That's one example. Second example, the mutation in the drug metabolizing enzyme determines how much of dose is required, right? And whether the drug is suitable for the therapy. We looked at clopidogrel And there are three set of populations. One set is poor metabolizer based on the cytochrome P450 gene mutations. Other one is intermediate, other one is normal. The 10,000 individual shows 15% are poor metabolized. That means they don't give any therapeutic response at all. 45% intermediate. That means 60% of the population will not get enough of benefit out of clopidogrel. But blindly if you take, it's not going to give any therapy. If you look at the economic burden, this is a survey from one of the pharma company. About 10 crore people take clopidogrel every single day. And the annual expenditure, if you look at 185 crore annually we spend by taking clopidogrel. Now you can see that after spending 185 crore, 60% are not getting therapy. So now using the data, genomic data, we can actually design a drug which is of haplotype specific, and I can't say individualized drug, but if you do overall all this 4,600 population, we may get some 10 different haplotype which is very effective for developing a drug. So we can have 10 different drug based on the genotype. So everybody will get therapeutic response. So this is, I think it's a little long, but for make you to understand. The second one you asked about pediatric rare disease. Rare diseases, one rare disease I said, when you look at globally, it's ultra rare. But if you go to a particular population, it's highly prevalent. Right? This pediatric rare disease program, when I was in CDFD, I initiated that. That look for a disease where so far no genetic mutation has been identified. Because the clinic and the genetic lab where they interact and they look for a specific mutation. They don't go for whole genome or whole exome. They screen based on the phenotype, okay, and wherever there's a mutation, then they settle there. But wherever they don't find mutation, then the sample comes to CDFD. We do the exome sequencing or whole genome sequencing. And always the novel genes will go into the procedure which I said, prediction, then animal model and so on. So once all of these are there, once we identify the mutation, then the next step is counseling, and also developing drug is part of that, particularly the hematological disorders. Right? So these two are very important projects funded by Department of Biotechnology. And the 10,000 genome is available now for anybody to make use of it. But there's a procedure to apply for that at IBDC, Indian Biological Data Center. Hope you got that.
Yes, sir. Thank you, sir. That was very beautifully and thoughtfully addressed. Thank you so much, sir.
Thank you.
Greetings to everyone. I am Darshini from Keshav Memorial College of Institute of Commerce and Science. I am a bit nervous, so it's the first time.
Relax.
I want every one of you to answer this question. My question is to, you have very vast knowledge, you have traveled to places, and but the first mindset that you had after the first graduate, what do you want to endorse in the people from our generation? That what do you endorse more, the patients, or what was your mindset when you first graduated, and what was the first step after, like, in which did you join?
I realize the question. So you are asking that after completing undergraduation, so what postgraduate you have to take? Was the question?
I would like to repeat, sir. No, what was your mindset?
Mindset.
Mindset that you want to endorse in the people from here. What do you want to endorse more? From which mindset did you had at that time? like patience, like that one I want to ask.
I think what you are saying is, what was our mindset when we did our undergrad program, when we completed our undergrad program, which has helped us to be what we are today?
Yes, ma'am.
You will be very surprised that my mindset was very bad at that time. So I was, yeah, I mean, it's not always determined. Your future is not like determined by your mindset. But one thing I will tell is that at any point, whatever you are doing, you do it with happiness, you do it with love, and you do it with focus. Like you, and one mindset that I had, probably that has helped, I don't know if it has, but is to continue and to be able to get the highest possible degree. And that's all I was interested in. The undergrad, I wanted to do post-graduation. And when I had choice of doing post-graduation, I did not want to do anything other than related to medicine. So I wanted, my thing was very clear that I wanted to be in the field of medicine, at least closer to the field of medicine. So I chose topics and I did work in that area. So first you have to know what you like, and then you stick to it, and you get the highest education possible in that level. And then your personality, as you mature, you will realize that if you are a person where you feel that, oh, I have to do something for the society, then you do it in a different capacity. Either you go into education system, you go into a research field, or you become an entrepreneur. Everything you will do will earn money, no doubt, but it should also give something back to the society. So you attain good education and give something back to the society. If you have that in your mind, along with making good amount of money, it helps.
Thank you, ma'am.
So enough, or you want me
No,
to
no,
say?
sir. Please, sir.
Okay, if you ask me, basically I am from a rural place. I am from a village. I don't know how many of you are from the village, right? Many, right? And my parents were farmers. Okay. And I studied BSc Zoology. I don't know how many of you are zoology. Right? Yes. Then MSc also in zoology. Right? Then I specialized in genetics. The reason is, living in the early life in village actually helps in identifying genetics as a field, right? You see the plants, whatever the school teacher teaches, but you can actually see that in your own field, right? The color transmitted to the next progeny. Then the cattle, also the skin color, coat color, and even the horn, same as the mother, right? So all of this, that actually motivated me to take a genetics. Okay. And I also have little bit skill in doing practical. And in fact, I used to take for the zoology school, frog and rat collected and take it to classroom for the practical. So with that, I thought I want to do some research at the time. Okay, that is all I carried. And once you get into PhD, of course, that whatever comes, because we don't generally at that point of time determine, but we have to keep options, whatever the best option available. We have to, in fact, I did not go for PDF at all. There was no PDF. When I was doing PhD, I got the job, so I directly came to CCMB. So if there is an opportunity, I would have gone. But this opportunity came, so I came here. So we keep the option open, and hard work is very, very important. I always say hard work is very, very, very important. but I don't know how many of you know that I used to work in the lab, even CCMB, job I don't consider as a job because research is not like 9 to 5 job. You have to spend a lot of time in the lab. Until COVID, I used to stay in the lab. After dinner, I go back, work till 1 o'clock, sometime 2 o'clock, sometime early morning I go. Okay, so that kind of time you have to spend in the lab. If you are really interested in research, you have to compromise many things, including your sleep. Okay.
Even I should tell? Yeah. I am from BSc BZC. So I got a seat in MSc Zoology and also in genetics. So I decided to go for a medical so that there I can interact with the patients. So I have chosen MSc Genetics. After joining the MSc Genetics, I have developed interest in immunogenetics. So I decided that if I do research, I will do only in the immunogenetics. So during my period of MSc, there in our department, only one professor was doing the research in immunogenetics, Professor Ishaq sir, but he did not have any projects to guide us. So then I looked out whole Hyderabad where the immunogenetics research is going on. So then I came to know at Mahavir Hospital, Dr. KJR Murthy, he was a clinician and he was working in the immunology of TB and asthma. So before that I have approached Ahuja sir also, who was doing cytogenetics, but I was not interested. So I approached him, then I applied for a project like Lady Tata Fellowship. I got a Lady Tata Fellowship, Junior Fellowship. With that, I have joined that lab and I was very much interested in research in immunogenetics. Because there I used to get a chance to interact with the patients. So I have done a lot of work at Mahavir. I completed my PhD there and I worked as a scientist till 2013. In 2013, again I got a chance to go back to our department for teaching. There, when I got a chance, I want to go back. Then I went back to the department and I'm doing teaching plus research over there. And very much satisfied with the work, what I'm doing. Still, I feel like doing more and more. What enthusiasm at this age I have, my students, I will not see in them. Okay?
And also don't compare with the IT professionals. I mean, the AI is going to take their jobs. Your jobs are safe. Just do what you want to do. And also family support is very important. Tell your family what you like, why you like, and what you do is so important for society. And I'm sure then they will start supporting you. Don't be disappointed. There is money. It's not like you have so many options I talked about with bioinformatics, data analysis. Just find your skill, whichever suits you, and pursue it. Pursue it with patience and don't compare. And I'm sure you'll all do very well.
My question is to Sumanlatha Ma'am. Good afternoon, Ma'am. I'm Bhavna from Koti Women's College.
Yeah.
My question is, are there any genetic reasons for weak immunity?
I didn't get it.
Are there any genetic reasons for weak immunity?
For baby?
Is there any genetic reason?
Malnutrition, like if your immune system is strong, you can fight against any of the diseases. So immunity, first of all, you have to develop the immunity. You have to keep your immunity strong, then you will be healthy and you can prevent any of the diseases, like infectious or any, which are the common diseases.
Yeah. Sometimes when we make dosa pindi, I just remembered that our mothers, grandmothers, they would make it with hand. So that will obviously improve immunity because, the bacteria and your so being very, very closed is also not good. At the same time, exposure to pathogens is also not good.
Yeah, as a geneticist, I always believe that for everything there is a genetic basis, right? So for particularly immunities, HLA has multiple copies, which gives kind of immune, and the way we expose our early age gives matter immunity, because you're already building your immunity, right? And as I said, being born and brought up in the village, I feel I have better immunity than my children, right? So they feel, get cold very easily, but I am the last one to do. I don't advise everybody to get at the initial early age any kind of infection, but it is naturally built. And this is also true in case during the COVID time, Indians have better position compared to others who have much better hygiene because of our early adaptation.
So, good morning, sir. I am Harshita from Aurora Degree College, sir. So, you said about cardiomyopathy disease, sir. You said that in mostly in South Asian populations it is seen. So, was it also seen in the North Asian population? And if not, what could be the reason for that?
No, disease is common across the world. But what I said, that particular mutation is present only in South Asia. For others, even the same gene has multiples of mutation. So other populations carrying the mutation in some other region. Right? Okay. This mutation, the disease is common across the world. But the mutation is specific to India and South Asian countries.
Okay, sir. Thank you, sir.
Maji.
Mr. Anuradha Ma'am, ma'am, how do we opt for genetic counseling career? And what is its scope nowadays in this era?
So courses are available, offline courses are available. I think you have to pass certain exams to be able to qualify. Doing a course in genetic counseling takes you, like, I feel 10% only. What you need is proper experience in a setting, either a hospital setting or a laboratory setting, for at least a couple of years. Then you will become a better genetic counselor, because every counseling is different, every family is different, every case is different, and you will have to have good amount of experience to become a good genetic counselor. But that's an excellent field to choose, and if you have good communication skills, both oral and written communication skills, and you have empathy for patients. It's like, I have seen many genetic counselors who say, you married your cousin, no? What else can you expect? That kind of sarcasm doesn't work. You have to have empathy, you have to be nice to patients, you have to understand where they are coming from, you should have patience. So if you have all those qualities, it's an excellent career to be in. Especially now, there are so many opportunities.
Yeah, to add this, you can sit now. So considering the number of diseases which are in India, particularly the population-specific diseases and common diseases, we need more counselors. We don't have many counselors. So I know in Hyderabad, in Kamanini, Dr. Annie Hassan, she has started this course. If you have time, you can go and talk to her. They have credit system, and they also have organization which organize conference every year. Okay, so I think probably you can look into the website called, I think, Board of Genetic Counselors of India, right? So you can go to Kamineni, Dr. Annie Hassan is the name, you can meet her and talk to her.
The Institute of Genetics also, they are doing diploma courses. Yeah. Even at the Institute of Genetics, Amirpet, they also have some diploma courses. You can find out from them.
You give to students who are interested in pursuing a research career.
Sir, you may want to answer.
Based on our experience, I would say two things. One is your focus, whether you really want to do research So that gives, yes, you wanted to do research. Second one, hard working. Without hard work, you can't really do much of research. It will be like normal. If you really want to make difference, and if you really want to make difference in a particular field, and now, unlike previous years when we were doing PhD, now there are a lot of materials available online. You can check something you are really interested, you see whether somebody is doing that work parallelly. Then it's a duplication, right? And see that you have a very fresh idea which has not been done earlier, then take that idea and develop your research program accordingly. But remember, hard work is important.
Good afternoon, sir. Why do children, my question to Anuradha ma'am, and the elderly people get so easily, TB gets so easily.
Repeat, ma. Relax, repeat nicely.
Why do children and elderly people get TB so easily?
See, TB is two things. Like children, if they have a low immunity, usually the children, I have done a study from 0-day BCG vaccinated children. I have monitored till the older age people. So we feel that when the child takes BCG vaccine at birth, and he has immunity. So once he starts going to the school, the immunity becomes low with the exposure of the non-mycobacterium tuberculosis. So there is a possibility that they may get a chance of developing a TB. But the studies have shown that at the age of 12 years, if there is a possibility of giving revaccination with the BCG, they'll have a better immunity. Okay, so at the age of 12, they'll be vulnerable towards many diseases. Like their immune system, there will be a change in the hormones. So there is they were trying to understand, and they were trying, in our papers also we suggested, at the age of 12 years, there should be a revaccination. So ultimately what I can say is exposure, and you should have a strong immunity to fight against the disease. Okay, older people also, you know that they'll have a weak immunity, and also there'll be stress. I was telling you so many risk factors for the development of the TB. So that could be one of the reasons for the elders also to develop the disease.
More immunity compared to other foreigners.
Yeah, because we are exposed to a lot of pathogens and we are more exposed to many of the bacterial and viral things. So that is why our body is resistant compared to others, where in foreign countries they won't use the immunity because it is they are devoid of all these bacterial and viral these things. So that is why there could be a difference.
But TB is common in India, right?
Yeah, it is more prevalent in India. Because I was telling you, malnutrition, poor ventilation, and you have to have you should not stay in this. Mostly I have seen in the slum areas. And also now the people of the middle income population also have, because we are very conscious about the diet and all. But we should have, first of all, I feel that you should have a proper meal and balanced diet, then you can fight against the bacteria. Because we are all got exposed to the Mycobacterium TB. Yes. Some may have, it will be in the latent form also. So whenever our immunity goes down, there is a possibility of developing the disease.
Ma'am, why is adaptive immunity memory cells not helping in TB
Because it takes its own time to act. You have seen in the corona also. They take its own time to act. So that is why it gets delayed.
Thank you, ma'am.
Thank you all.
Sir, what's the truth about Aryan invasion and that Aryan descendant population in the Himalayan region like Ladakh and Kargil?
Yes, brought the topic. In fact, one of our study, we clearly shown there was no Aryan race existing as a unique in India. Because you don't see when you do the analysis, there's no distinct cluster. If you take all Indians, only Andaman is because they are in isolation for a very long time, they have unique set of mutation. Only they are away from the rest of the clusters. Whereas the rest of the population, they're all admixed. There is no specific population exist as unmixed. So that theory we have already demolished. There is no Aryan invasion. Ladakh. No, that probably somebody must have written based on some history, but genetically there is nothing to prove. After the first out of Africa human, there are subsequently several migrations. Jewish, then we have Siddhis, many populations who have migrated, and all of them have admixed, including the recent one is Siddhis. Siddhis also admixed. In fact, one of our study using the bone samples collected from Western Asia, it shows that there was a migration from the Central Asia towards Europe and also towards northern part of India. That was around 1000 to 2000 BC. It's not very old. They are the one probably brought the Indo-European language to India and also to Europe. That's also not very old, 1000 to 2000 BCE, and they're all admixed. There's nothing here existing as a separate population where we can call as a Aryan.
We'll move forward. We now request Dr. Rajender Rao, a scientist, CSIR, NIN, to present the concluding speech. Oh, sorry, ICMR, Nutrition.
Afternoon. Say good afternoon, no? That's good. Now, time for the lunch. I am also feeling hungry. The biological clock will tick at 1 o'clock. Sharp we eat at 1 o'clock. But sir said, in research it is not like that. We eat at 4 o'clock, 3 o'clock, depends. So, I would like to thank the organizers for bringing people together. So, the DNA dialogue. So, my congratulations to the organizers, especially the Aurora Degree College Management and the Padma who is back of me. Sir, I want your presence. That's what I am here. Right, so just I came when the dialogue started, the question hour and all. Yeah, the DNA dialogue. Myself is a geneticist. I did my MSc Genetics in Department of Genetics, PhD in Genetics, and the journey in the genetics. So I missed the chemistry, that's what I joined in genetics. Someone was asking the question, no? So to be frank, my passion for the chemistry, till I speak the chemistry, I am good at the chemistry, I read the chemistry, I read the chemistry, I read the chemistry. But the thing is, whatever we choose, we have to excel in that. There is only the success, nothing, the mantra, madam told, sir told the same thing. Everyone will tell that only. We have to have that dedication and all. Don't look and compare ourselves with the AI people and all. The DNA dialogue is that now 3 billion base pairs are there. What are they doing in the body? A question comes always. Only 2% is a functional. We read the literature and all. Remaining is a junk, what we call. No use, nothing. Why nature selected that much of the DNA? Why 23 pairs of chromosomes are required in the body? The question comes always to me. So there is a lot to do, go for the journey. That's what no, today, so when Anuradha Madam is saying, no, I started the company, there is no genetic company and all. Now, within seconds, we can sequence and we'll get it. When we started our things and all to see the PCR, that was in a small room. So it is not allowed to see the PCR. Now we can carry the PCR in hand. So that was the thing. So being a conventional geneticist, so we studied the only conventional genetics, how the Mendelas and all, how the mutation has happened, how it is passed to the generation to generation. These are the calculations. So what we did is in population genetics and all. Now the thing is evolving very fast. Are we really adapting? How many of you are reading the books? The question comes that the 3 billion bases are doing. What are they doing? Always a question is there. M13 is also living, the virus, just 1300 base pairs. Recent time corona has come, a small size. So there is a lot to go for it. So then the DNA dialogue, lot many things are there to look into that one. So when it will come for us, when we bring this type of the meetings are very much important to interact with the people. That's what, no, again, so give a big round of applause to the organizers. So then, not a joke. So behind it, some hard work is there, right? Whom we have to select and all. So they selected the eminent personalities. One is from the academy, one is from the research, one is from the company. The 3 different sets of people are here. So patiently they are giving, thank you very much, sir. So it is a really, so see Thangaraj sir to go to the CCMB, it was a hurdle for us that time, believe it or not. When I started working with the Thangaraj sir, I felt that how nicely this is. I learned many things. It happened to be in the last year, we gave a talk in the Kerala, right sir? So myself and sir is on the same dais. So this comes when your passion, your dedication, all are important. AI era is coming. The AI era, AI is a small tool. I always believe that is a tool. It is developed by us. How to use it, we have to know. But I want to be clever than that because no ultimate decision is by me, not by AI. So that no, in genomics, can we do how to use in the genomics? How best I can use? Now, my journey is on diet gene interactions. I am working on the nutrition lab. Nations of nutrition. How the DNA and the RNA, so which are interacting with that, how it is changing the dynamics of the DNA methylation. So how to use the technology to get for a conclusion. Sir said, no, simply we can, earlier it was our days to do all the things, now we can do in a minutes. After that, no, it will give some data in front of us. Then interpretation is finally by us, not by the AI. We have to. Then it will come when you read the subject. Are you really reading the subject? Yeah, confusion state is there always, the BSc to MSc, MSc to the PhD. After the PhD also a confusion is there. What to go, where to go. When you have the subject, hard work and your dedication, definitely will show the way, the money will follow you. Definitely it will follow. For that, no. So sir started, so connecting the dots. His journey, you heard his lecture, I heard 2-3 times. It is a very fascinating. Whenever I listen, it looks very new for me. Because no, the journey started from just now, someone asked from the, so Kashmir to the Kanyakumari and Andaman. It did not leave anyone. How to connect all these things? So that no, that we have to connect the dots. So shall we? The question comes for us. So there is a lot to do in the genetics to understand that. So the conventional genetics has started where we started calculating the checkerboard we used to put and all. So maybe Sumanlatha knows, I was from Telugu media, I used to take their help to understand in English. So then what is this checkerboard, what is mental, what is the nuisance here? Run, leave the subject and go again the chemistry. Then Professor Vaidyanath, sir, this is thanks to him, he hold my hand, no Rajendra, you be here and prove yourself. So someone will be there, we have to listen to them. That is important for us. So then for that what hard work I have to put. Really believe it or not, I don't know about the dictionary. He has given me one Lifco dictionary, Telugu to English, where we can start the things, the journey and all. The fashion to listen, we can see the statics and all, we was having the J. Smurti sir. So sir lecturers went on well, so for 4 to 5 hours one day, afternoon session, after the lunch, very interesting. That type of the teachers we were having. Second is, then we have to have our own fashion to learn. Now complexity is there, simplicity is there in the genetics. So now the dialogue is very good. I am really enjoying. I learned many things. So by listening all the things, every day is a learning. Being a researcher, being a teacher, being a company person, if we don't adapt the latest technology, latest happenings, we don't go through that, we'll be back always. People will push back. We have to read every day. There is no other go. You think that only the computer people, they have to upgrade every day? No. Everyone has to upgrade. We have to upgrade. For that, no, every morning, Yahoo and all the WhatsApp is secondary first. Then I'll open the PubMed, I'll put my topic and I'll see what is happening in that. How many papers, new papers have come. Then generally I put in the Google and see what is happening in my area of research. So what are the changes are happening? Then how I have to go forward, I have to take it forward. The idea, whatever I got, I am working on that, so I am taking forward. So for that, what I can add? Of course, our students are there, my PhD students are there. Besides, no, having the knowledge is important for us. Then second, so to start this now you are lucky enough, when madam started the company, I know how much hard work she took and all the licenses and all. Now the incubators are there. Everywhere, many incubators are there. If the idea is there, there is a money is there. People are there. Now only thing is, you have to read the books, you have to come with proper idea. What do you want to do? Just I want to start a company, my dream is to become a geneticist, no. Yes, dream is very much important. So that no, Abdul Kalam says, dream big. Yes, for that we have to put the step in proper way. Are we putting the steps? It was started somewhere, they said the factor, 1902, Gerard theory explained that one. Something happened, that's what no phenylketonuria we got it. There is no word of the genetics, they said the factor. After that the Ingram, they mentioned that no one gene, one enzyme story, so where it is connected the things. Now we know what is the problem. We know the sequence, we know the dot. So that know how it is evolved, then we have to evolve with the times. Then you have the time, then we can do. The many ways are there to go for it. From genetics, the variation is there, the phenotype is there, the genotype is there. Always we can see only phenotype, how we look. But there is a genotype is there. Everyone is having the tremendous knowledge in you. That is the genotype. You underestimate yourself. We compare with others. No. No need to compare. You have your potentiality, you have your fashion, you have your own life. For that what we need is, nicely they told all their experiences, really great. So that from we have to learn, and also the dialogue is a DNA dialogue, not only the dialogue, carried dialogue also I saw here, and also the way forward they showed. These are all important for us. Really I am also enjoying by sitting there, so then how we can evolve further. Sir, what is this, sir? You are all grown, you are having a lot of experience. No, still I am learning. I said, no, AI. I learned from my child, believe it or not. He is a computer fellow, and also my students who are having the more knowledge in that area. I can sit with them and I can learn. What is AI? What it is doing? So I read myself, then how can I put this thing to my work? So can I draw some conclusion from that? Can I take help of that one? It is my assistant, a nice assistant, a smart assistant, a multitasking assistant I can say. It is a multitasking assistant. A tool. If you adapt the technology, if you understand the technology, then we can. So one thing I am noticing, nowadays we are killing our time most of the time. Sir said, hard work is important, sometimes we have to forgo our sleep also, yes. Now, no, we are losing our sleep because of this. More shorts, more unnecessary things we are saying. The same time, I want to say, if you put on your education, your subject, definitely the DNA dialogue, whatever the way you came here, all the way to attend this one, these three eminent people are here, their interaction and all, definitely it will give a meaning for you. Once again, thanks to the Aurora College for bringing the people together and organizing this type of things. So thank you, Padma, for inviting me. Thank you. Wish you good luck. As I am from the National Shop, the nutrition, I have to disseminate the knowledge of our things. So how many of you are having the habit of eating the fruits? So seasonal fruits or vegetables are very much important. We have to eat it. Don't ignore this. Because now immunity word is coming. During the corona time, everyone is shaken. So how to get the immunity? Then started eating. Now also we can eat. Every day we can eat. They make the promise yourself that every day I eat the fruit and the vegetable. Don't ignore it. Seasonal fruits and vegetables are very cheaper than the whatever the you are investing on this for your talking and all. So this is a diversity in the diet is important for us. This is a message from the NIN. Always we see your health is our country's health. You want to see the Vikasit Bharat? Mera Bharat? Say loudly, Mera Bharat. Yes, we have to make it Mahan. We are traveling through this one. We have to see the Vikasit Bharat soon. It is a developed country soon. All we have to work together. So this is all our duty to do. Okay, thank you, Jai Hind. Thank you very much.
Thank you so much, sir. Now I request our principal, Dr. Ajay Kumar Garu, to felicitate our speakers.
Thank you so much, madam. Now I request Rajendra Rao sir to please come forward. Now I shall hand over to Padma madam to give vote of thanks.
Over to you, ma'am.
Thank you, Silpa. Distinguished guests, participants, and my colleagues, it is my privilege and honor to propose vote of thanks. We are eternally grateful to Dr. Kumaraswamy Thangaraj sir for his time, insights, and invaluable knowledge shared with us. I profusely thank Dr. Anuradha Udimudi garu, Dr. Sumanlatha garu for sharing their knowledge, research for making this session meaningful. We are indebted and grateful our Honorable Secretary, Dr. Ramesh Babu Nimaturi garu, for his benevolent consent for conducting the talk today. We are deeply grateful to our principal, Dr. Ajay Kumar garu, for his guidance and unflinching support in organizing the talk today. We are much obliged to express our gratitude to Vice Principal Sai Krishna garu, Hari Prasad garu. We are also indebted Dr. Rajendra garu, Dr. Srinivas garu, Dr. Venugopal garu, Dr. Sanath sir, Dr. Pandari garu for their gracious presence today. We express our gratitude to our colleagues Dr. Sharath, Dr. Vasudevan Ranganathan garu, Dr. Nagavani, Dr. Krishna Subha for their invaluable contribution, guidance, and support to us in conducting the program. We are also thankful to all the distinguished faculty from several colleges of Hyderabad and other regions who graced this program. We appreciate the work of our staff, technicians, and volunteers who managed all the nitty-gritty. We are all thankful to all the participants of their active participation and engagement. We leave here today inspired and transformed by the new knowledge shared and connections made. Thank you all for being part of this successful event. Thank you.
Now request to collect the certificates to all the participants. Respected college or one representative from the college can take all the certificates if possible.